एग्जाम भी है ना।
पोस्टर
पे अगर हो गया तो इधर
ठीक है।
Good morning, Jasmina ma'am. Your voice is not audible. I think so. Your mic is mute.
Good morning, ma'am. Good morning, ma'am.
Good morning.
Good morning, madam.
Good morning, sir.
And conquering it. We enable you to break the old. We are the Gujarat Technological University, the International Innovative University.
A very good morning to all dignitaries, guests, colleagues, and participants. I, Dr. Jasmina Surti, on behalf of Shri Narjibai Lalbhai Patel College of Pharmacy, warmly welcome you to the one-day virtual 12th Indo-US conference on theme Nanotechnology in Pharmaceutical Sphere, Contemporary Approaches and Pharmaceutical Applications. Organized and hosted by Shri Nanjibai Lalbhai Patel College of Pharmacy in collaboration with APP Gujarat State Branch, APP American International Branch, APP Drug Design Medchem Division on January 30, 2021. We will start our program with remembering the Almighty to bless us for successful completion of this program. For this purpose, I request all of you to join the prayer session.
तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमा आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदाम् जाड्यान्धकारापहा हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्
Thank you, all of you. Now, let me say about our institute, Shri Nanji Bhai Lalbhai Patel of Pharmacy. Our institute, Shri Nanji Bhai Lalbhai Patel College of Pharmacy, was established in the year 2004 with Bachelor of Pharmacy course. At present, we offer B.Pharm course and M.Pharm course in pharmaceutics, pharmacology, and quality assurance disciplines. Our institute is approved by AICTE and PCI New Delhi and affiliated to Gujarat Technological University, Ahmedabad. We are having a great facilities like well-equipped laboratories, well-maintained classrooms, library, hostel, and transportation facility. Our institute has always provided its students with a healthy environment to help them grow in every sphere of activity, to be its academics, sports, or any other extracurricular role. We had organized many guest lectures and seminars at our institute to provide students with good knowledge. Now, the Association of Pharmacy Professionals APP is a unique organization for more than 1,200 dedicated personnels willing to serve pharmacy profession in dynamic, which involves people from various arenas, including students, teachers, researchers, scientists, as well as industry professionals. APP is continuously gaining attention among pharmacy community of India and abroad. APP actually aids in extending the knowledge and facility of several technical institutes, industry as well as health sectors to the remote areas where service and life functionality of pharmacy profession is the real bond. Today, it is our immense pleasure to have presence of dignified personality. To begin with, Shri Kirit Bhai Patel, Secretary, Vidya Bharati Trust, Umrak, Shri Ashwin Bhai Patel, Joint Secretary, Vidya Bharati Trust, Professor B.N. Suhagya Sir, Chief Advisor, APP Gujarat State Branch, Professor S. Narasimhamurthy, Vice President, APP American International Branch, Professor Vannabi Patel, President, APP Gujarat State Branch, Dr. Rajiv Dahiya, Organizing Chairman and Founder, President, APP, Professor Dhiren P. Shah, Organizing Secretary and Principal of Shri Nanjibai Lalbhai Patel College of Pharmacy, and Professor Biren Shah, coordinator of this conference. I welcome all of them on behalf of Shri Nanjibai Lalbhai Patel College of Pharmacy. Further, I would like to inform that our today's conference is currently live on Facebook as well as on YouTube, so all the participants can access it. Today, we have with us Professor Dhiren P. Shah, Organizing Secretary for this conference and Principal, Shri Nanjibai Lalbhai Patel College of Pharmacy. Sir has more than 20 years of teaching experience, has published more than 50 research articles, has presented more than 15 papers in national conferences. Sir has contributed 4 books and has received grant of around 2 lakhs for various projects. Sir was selected as mentor for International Exchange Program of GTU with Laurentian University, Canada in 2017. Sir is Associate Dean for Zone 5 of GTU and member of Board for Studies for Department of Pharmaceutics. Now I request Professor Dhiren Shah to address the participants. Please, sir.
Yeah. Thank you, Jasmina Madam. A warm welcome to all of dignitaries, participants, dear faculty members. It is my great pleasure to welcome you all in a one-day 12th Indo-US virtual conference. We are grateful to our Managing Trustee, Shri Manarbhai Patel, Secretary, Shri Kiritbhai Patel, Joint Secretary, Shri Ashwinbhai Patel, other trustee members, Shri Hasubhai Patel, Shri Bharatbhai Patel, Shri Mahendrabhai Patel, for their tremendous support they have provided to us to organize this particular conference. I will say few added words about our college. Our college is run by Vidya Bharati Trust, Umrak. Sri Nanjibai Lalbhai Patel College of Pharmacy, Umrak, established in the year 2004, and year by year the progress of this institute is achieving the newer heights. In 2010-12, the M.Pharm was started with the different disciplines, and presently The MPharm is available in a 3 branch. This our college is already applied for this accreditation. We have doing very good research activities as well as the placement for the students. So this is about the college. We are proud to announce that this conference is being conducted along with this collaboration of Association of Pharmacy Professionals. As a host and to the conference organizing committee, we would like to express our heartfelt thank you to the Association of Pharmacy Professionals who have given me this opportunity to organize this event. I welcome Dr. Rajiv Daya sir, organizing chairman and founder, president APP, Dr. Bihen Suagia sir, chief advisor APP, Dr. Sunita Daya, general secretary APP, Dr. Vandana Patel, president APP Gujarat state branch, whose efforts bring this conference at our institute. So I am really thankful to madam. Dr. Ankit Chaudhary, Vice President APP Gujarat State Branch, Dr. Mehul Patel, Vice President APP Gujarat State Branch, and Dr. Nehal Shah, Secretary APP Gujarat State Branch, whose members have put together and engaging this program. The pharmaceutical industry plays a pivotal role in keeping the world populations in good health and ensuring that they have a better quality of life in India. The pharmaceutical industry has contributed significantly not only to the health security of our people, but also to the economic growth of our great country. Over the last 3 decades, companies have increased availability of medicines by 50% and thus have contributed to reducing the disease burden by 36%. The cost of the treatment of for the life-threatening diseases such as chronic leukemia, hepatitis C used to be a prohibitive earlier, thus making it unaffordable to many. But today, these costs have come down due to the original treatment cost, thanks to all progressive measures taken by the pharmaceutical industry, pharmaceutical educational institute as well. No doubt that this country has brought and inspired significant changes in the field of pharmaceuticals. These changes are based on current research with ramifications on students, teachers, and educational institutions. As you know, there are a range of reforms under consideration in the field of nanotechnology also. The speakers in this program are uniquely placed to debate these and highlight the key themes, trends, and the current practices for the audience. Especially interesting will be to learn the priorities and the development from the several local, regional, and international organizations, companies, standard setters, and other influence. This is the first international conference in the history of our college, which is completely conducted in a digital platform in this COVID-19 situations with maintaining the social distancing and other protocols. The theme of this international conference is the educational dynamic due to the COVID-19 pandemic. I welcome to all eminent speakers, Professor Alikha Das from USA, Dr. Sushma from New Delhi, Dr. Atindra Shukla, DDU, Nadiyad, Dr. Palaniswamy from Tamil Nadu, and the guests from all over the country, from the different walk of life. You join here to share their knowledge and vast experience with students, teachers, and scientists. It is my pleasure to welcome all participants today for this one-day digital international conference. My thanks and appreciation also goes to my faculty members, non-teaching staff members who have worked without any tiredness and show their outstanding enthusiasm. The year ahead looks promising and we hope to sustain the momentum that we are carrying since 2004. As far as the conference considerations, we secured the more than 300 number of participants in a short period of time and more than 150 posters in this conference. And I am really thankful to my all gurus, particularly Dr. Suhagia sir, who has helped me a lot for this organizing these events and for the such type of this, the participation within the notice, within the short period of notice. As well as I am also highly thankful to the Dr. Vandana Madam, Dr. Ankit Chaudhary for giving me a very good support in terms of the participants as well as for this the abstract. Again, I would also like to thankful to Dr. Yom Upadhyay and Dr. Chainesh Shah from the Sigma Pharmacy College for the more number of contribution in this conference, as well as my colleague Dr. Vinit Jain. He has also contributed a lot for this conference. So I welcome all this on this digital platform. While rushing to answer is not what academics do, we hope that this conference generates a lively discussion as well as raise more questions about how we can go through about the nanotechnology in 21st century. I would like to conclude my speech by encouraging the delegates to participate with an increasing number in all the conference through the digital platform. I wish everyone a successful, safe, and fruitful conference. Thank you and enjoy the conference. Thank you.
Thank you,
Jasmina,
sir.
madam.
Thank you, sir. We are obliged to have Professor Vanna Patel, President APP Gujarat State Branch with us. Dr. Vanna Patel is working as Professor and Dean Doctoral Studies and Research in Parul Institute of Pharmacy and Research at Parul University. Ma'am has rich experience of 29 years in academics in various capacities. Ma'am has guided 55 students and for MPharm and 5 PhD students for their dissertation. Ma'am is involved in industrial consultancy and co-guided more than 25 PG students for industry-defined projects. Ma'am is recipient of 8 funding grants. Ma'am has authored 3 book chapters and 1 book and has published 3 Indian patents. Ma'am has coordinated the organization of about 35 seminars, workshops, and conferences and has delivered 36 lectures in conferences and refresher courses, including one international conference. Her commitment to pharmacy and education field has been well recognized, and she is the recipient of prestigious awards like Shiksha Gaurav Puruskar Award in 2015, Innovation in Higher Education Award 2015, GTU Innovation Council Award in 2016, Principal of the Year 2019, APTI Woman of the Year Award in 2019, and many more. I welcome you, ma'am, and I request you to say few words to audience. Please, ma'am.
Thank you, Jasmina, for giving this nice introduction. And thank you for giving me the opportunity to interact. Good morning and good evening, good afternoon to all those who are on the screen. As a President APP Gujarat State, I, Dr. Vandana Patel, heartily welcome one and all for this one-day virtual 12th Indo-US conference. At the outset, I would like to congratulate Dr. Dhiren Shah, Dr. Biren, and his entire team, as well as the management of Shri NL Patel College of Pharmacy, Umra, Gujarat, for taking this enthusiastic initiative to organize this conference. I also acknowledge the support by Vidya Bharati Trust, Kirit Bhai, Ashwin Bhai, who are with us on the screen. The conference is based on the theme of nanotechnology, and I'm sure the delegates are going to be benefited by the experts who are expert in their own field and who are ready to share the knowledge with the students and faculty. I also thank Dr. B.N. Suhagya sir, Dr. Mehul Patel and team of DD University for collaborating with APP for this conference. We are highly fortunate to have blessings and support and guidance of Suhagya sir whenever we organize such event. Thank you, sir. Thank you for being with us on this platform. We are also indebted to Professor Alekha Das and Professor S. Narasimha Murthy, office bearers of APP American International Branch, for their all-time support and guidance. Association of Pharmacy Professionals under the able leadership of Dr. Rajiv Dahiya, Founder President, and Dr. Sunita Dahiya, Vice President, has shown good progress at a very good pace, and we, the pharmacy profession, is getting benefit of all the efforts that they are putting it into motivating the researchers, motivating the professionals, and highlighting the achievements and efforts of the professionals. Thereby, every one of us are, recognizing our own importance of pharmacy profession. So I thank Dahiya sir and madam for doing this service to the profession of pharmacy. I also welcome and thank all the chairpersons who are going to be with us for this conference. And I also thank my team of APP, uh, Ankit sir, Nehal sir, Mehul sir, Suhagya sir. Uh, in Gujarat state, we want to see that the APP, with the support and help of APP and the guidance of the senior people in our field, we in Gujarat state will do various kind of activities in time to come, and we will see to it that we can contribute to our profession of pharmacy in a better way. I also welcome all the delegates who have shown overwhelming response to attend this conference. And dear delegates, on behalf of APP, I assure you that the expertise shared by all the renowned speakers will definitely benefit you and will definitely help you to add the knowledge and to give different direction in your thought process, whether you are doing research or whether you are doing your graduation or post graduation. So please take enough benefit from their speech, from their expert talks, and you can be connected with us anytime. We are there to support you in all your future endeavors also. So I conclude my speech by giving the best wishes for this program. Thank you all.
Thank you so much, ma'am, for sparing your time for us. Now, we are honored to have dignified personality, Professor S. Narasimha Murthy, Vice President, APP American International Branch with us. Sir is presently working with School of Pharmacy, University of Mississippi, USA. Sir has experience of around 28 years. Sir has guided 8 postdoctoral research associate, 15 PhD students, and 5 master students for that dissertation. Sir has worked as editorial advisory board member for many journals and research grant proposal reviewer. Sir has received more than 25 research grants. Sir is chief editor for 2 books and authored many chapters in various books. Sir has more than 133 publications and 217 scientific presentations to his account. Sir is invited speaker for more than 30 seminars. With this, I welcome you, sir, and I invite you to say few words. Please, sir.
Thank you, madam. Hope you can hear me. Um, Hello, everybody.
Yes, sir, you are on.
Am I audible?
Yes, sir, yes, sir.
All right. Yeah. Thank you, madam. Thanks for the introduction. So, hello, everyone, and I welcome all of you, all the participants, speakers, delegates, organizers, everybody who's a part of this international meeting on behalf of APP, American International Branch. So I know quite a few speakers who are talking today. So I welcome all the speakers. I'm looking forward to listen to their enlightening talks as well. I really thank all the organizers from SNL Patel College of Pharmacy and APP Gujarat State Branch and everybody, and the speakers who gave the welcome speech initially. Wonderful welcome speech it was. I believe that what begins well will end well. So I'm sure this conference is going to be a very successful conference. And the topic is very interesting. I see that it is nanotechnology in the pharmaceutical sphere. Nanotechnology is definitely there in every sphere of pharmaceutical formulations, be it the drug delivery systems, be it the herbal drugs, be it anything that we do in pharmaceutical field is nowadays becoming nanotech, technovized. So that's why I think this is a very contemporary topic, and this is definitely a topic of interest to the youngsters, the researchers, everybody who are a part of this organization. So I think this is a very well thought of topic. I'm really hopeful that all the talks you will find interesting and you will find them useful. I hope the participants get a chance to ask as many questions as possible. Students must be there in the audience also. I encourage everybody to ask as many questions as possible, because all the speakers I see, they are kind of very well accomplished speakers. They have done a lot of work in their fields. They have done a lot of research in the nanotech area. So I'm sure that their talks would be very interesting, and they would be able to address your questions and enhance your knowledge in the field of nanotechnology. So I also would like to thank Dr. Rajiv Daya and Mrs. Daya for their tireless efforts in making all these conferences happen. I don't know like how many hours a day they sleep. They work really, really long hours and put all these things together, put all of us together as well. So it's really a wonderful service to the profession of pharmacy. So I wish them all the best. I wish all the participants and the speakers all the best, as well as the organizers all the best. So I hope this conference is going to be a very successful conference. I'm very confident about it. Thank you all for the opportunity to talk a few words about the conference and about the speakers. It was wonderful interacting with everybody. Thank you so much.
Thank you so much, sir. Now we have with us Dr. Rajiv Daya, sir. Dr. Daya is presently Director, School of Pharmacy, Faculty of Medical Science, the University of West Indies. Sir is President of Association of Pharmacy Professionals and Editor-in-Chief of many reputed pharmaceutical journals of international level. Up till now, Sir has published more than 70 research papers, review articles, and 3 book chapters with Springer and Taylor and Francis. He has been invited 23 times as a guest speaker in various conferences, including 37 orations and 105 poster presentation. Dr. Daya is honored with Most Productive Researcher Award in March 2019 by Dean Faculty of Medical Sciences, the University of West Indies. Dr. Daya has supervised or co-supervised more than 20 MPharm projects and 4 PhD projects. I welcome you, sir, and I request to reveal the objective of this conference. Please, sir.
Yeah, thank you for my introduction. So I welcome from bottom of my heart, especially to all of the speakers, all of the organizers, my APP office bearers, especially belonging to the American International Branch, as well as the Gujarat State Branch and the concerned division involved. I would like, before saying anything, I would like to start by thanking Sri Manhar Bhai L. Patel, who gave us chance to host the APP 12th Indo-US conference at the beautiful campus of Sri N.L. Patel College of Pharmacy, Bardoli. And obviously the efforts of Professor Dhirensa can't be ignored, because without the hard efforts of Professor Patel, it was not feasible. With the support of our president of Gujarat State Branch, Professor Vandana Patel, under the guidance of Professor B.N. Suhagia, who is the Chief Advisor of APP Gujarat State Branch, as well as Chief Guest of today event, and working as Dean Faculty of Pharmacy, Dharam Singh Desai University, Nadiad, Gujarat. So, if in terms of APP, we were a small family before 9 years. We started from 7 members with some certain aims and objective, and after 9 years, we are very glad to say we are running ahead. Every college where I say they are ready to host our event. They well understood our objective, our cause, what we are doing and or what we want to do for the profession. And I'm highly thankful to each and everyone who listened to us and giving their vital contribution toward the development or toward the further progress of our pharmacy profession. We stands with 8 international branches, and we have 14 national branches, 7 divisions, and 2 local branches. We are uh, desperately looking for our new branches in Kerala state, and recently we have launched our new division, which is APP Pharmaceutical Education and Healthcare Management Division. Um, Also, suggestions are welcome to start new divisions or new branches where we can do the best for our lovely profession. Regarding speakers, today we have involved 4 speakers. One of international community, well known. He is our president of American International branch. He is Professor Alekha Das, who is working as Associate Dean Research at Creighton University, Omaha, Nebraska. Very enthusiastic person, always want to involve as speaker, and I'm highly thankful to him. Even if I ask once, so he's ready to deliver. So thanks for your time, sir. We have 3 national speakers of repute. First one being Dr. Susma Talgaonkar, who is working as Associate Professor at Department of Pharmaceutics, DPSRU, New Delhi. She is a very active researcher and a very sound publication profile, and she is serving presently as the Vice President of APP Delhi State Branch. Our third speaker is Dr. Atinder D. Sukla, who is the Director in Charge and Associate Professor at a Nanotechnology Center at DD University, and he is recipient of our Young Scientist Award. Usually we deliver this particular award those who have attained a ISI impact factor of 100 and above. Our last speaker is Dr. Selvamani, who is Secretary of APP Tamil Nadu State Branch and presently working as Associate Professor at Center of Excellence in Nanobio Translational Research, Anna University, Tiruchirappalli, Tamil Nadu. So he is also expert in nanotechnology field, and we try to keep all the persons who have expertise in nanotechnology. But that does not mean our conference covered only nanotechnology. It also covering several sectors of each potential pharmaceutical area, including medicinal chemistry, drug design, pharmacology, pharmacognosy and natural product chemistry, where all have submitted their posters in form of e-posters, and they are being evaluated. So with these, I would end my speech, and I would extend my thanks to all the parties involved in the organization of this APP 12th Indo-US conference. Thank you very much.
Thank you so much, sir. Now we have with us Professor B.N. Suhagya, sir, Chief Guest and Dean, Faculty of Pharmacy, Dharamsi Desai University, Nadiad, Gujarat. Sir is an academic with vast experience of more than 44 years in academic at graduate and postgraduate level. Under his able stewardship, 35 students have been awarded doctoral degree and 40 students have been awarded postgraduate degree. Sir has published more than 190 research papers and has presented more than 234 research papers in national and international conferences. Sir has published 2 books and has received 11 awards for research presentations and APP Outstanding Achievement and Best Researcher Award. Sir holds life membership for many governing bodies. Now I invite Professor B.N. Suhagya, sir, for inauguration remark and opening of scientific sessions. Please, sir.
Okay. Thank you, Jasmina, for a nice introduction. A very good morning to one and all. Respected Sri Manarbai Patel, patron and trustee of the Vidyabharta Pharmacy College, Umrah, Kirit Bhai and Ashwin Bhai from the management, Rajiv Dahiya sir, Sunita Dahiya, Professor Vandana Patel, President of Gujarat APP branch, Professor Narsiha Murthy, Vice President APP US Branch. Speakers of the event, Hiren Shah, Organizing Secretary, Dr. Nehal Shah, Secretary, Dr. Mehul Patel, Vice President, Dr. Ankit Chaudhary, Vice President, staff members of the college, and my dear participants. I feel privileged to inaugurate today's event of APP Gujarat and American branch in association with Dharamsi Desai University. I welcome all the members and appreciate the pain taken by behind it. I am thankful to all keynote speakers to spare their valuable time and willing to provide healthy and worthwhile brainstorming platform to all participants. I congratulate Dr. Dhiren Shah, principal of the college and organizing secretary of today's function, uh, to manage all very well within the short notice. Dhiren, keep your tempo with your smiling face. I congratulate Professor Rajiv Dahiya and Sunita Dahiya. I can see full dedication toward the strengthening of APP all over the world. I appreciate the working goals and readiness of APP Gujarat branch and their office bearers, say Professor Vandana Patel, Professor Mehul Patel, Professor Ankit Chaudhary, Professor Nehal Shah, Professor Samit Bhai, and all others who uplift pharmacy education. So herewith I am very delighted to open the scientific session and ending inaugural ceremony. I hope all participants enjoy this event. All the best for the event. Thank you. Thank you.
Thank you so much, sir, uh, for your words. Now, I am thankful to all the dignitaries for joining this inauguration session. We are glad to announce that Shri Nanjibai Lalbhai Patel College of Pharmacy has organized a webinar on how to crack GPAT on 4th February 2021 for students of 3rd and final year. We have also organized a free mock test for GPAT on 30th February 2021. The first 5 students who will secure highest marks will be awarded with cash prize. Yeah. Now, with this, we look forward for a successful conference, and I strongly encourage you to become actively engaged in various lecture sessions and discussions. Okay? We will be starting our lecture session in few minutes.
Thank you. Thank you, everyone, for joining this inaugural session for this conference.
Thank you, sir.
Thank you, madam. Thank you, Swagya sir, Daya sir, everyone.
Thank you, sir.
So, Dhiren sir, so should we go out and then we have to be in, or we have to be in continuation? Hello?
Sir, you have to discontinue, and you can join with the link of YouTube or Facebook. Okay, so we can in the speakers as well as chairpersons.
Okay.
Thank you, sir. Okay, so moving towards our lecture sessions, our first session will be delivered by Professor Alika Das, Carrington University, Omaha, USA, and Dr. Susma Telangankar, Delhi Pharmaceutical Science and Research University. For this session, we have with us Professor Govinda Sami Jayabalan and Professor M. Vijay Anandi to chair the sessions. Professor Govinda Sami Jayabalan is currently working as Principal, Alwar College of Pharmacy, Rajasthan. Sir is ex-Dean, Faculty of Pharmacy, Rajasthan, University of Health Sciences. Sir has 26 years of teaching, 1 year industry, and about 20 years of research experience. Sir has guided 18 PhD students and 5 students are in progress. Sir has convened many conferences, chaired many scientific proceedings, and actively participating in education. I welcome you, sir. Professor Vijay Anandi is presently working as professor and head Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Science, Wells Institute of Science, Technology and Advanced Studies, Tamil Nadu. Sir has total teaching experience of 21 years, research experience of 19 years. Sir has total 178 publications, among which 121 is Scopus publication. Sir has 2 patents to his account and has received fund of 28 lakhs 51,000. Sir has supervised 5 PhD students and 8 are ongoing. Sir has supervised 45 M.Pharm and 31 PharmD students. I welcome you, sir, on behalf of Sri Nanjibai Lalbhai Patel College of Pharmacy. So now I request Professor Govinda Sami Jayabalan to introduce our first speaker, Professor Alekha Das. Over to you, sir.
Good morning. Am I audible?
Yes, sir, you are audible.
Yeah. Very good morning, all of you. And I thank you for the opportunity to chair the session. And I wish the one-day virtual 12th Indo-US conference of APP become a successful event. Thank you very much. And without taking much of time, I would like to invite by reciting the profile of Professor Aleka Dash. Dr. Aleka Dash has received his bachelor's and master's degree in pharmacy from Jadavpur University, India, and a PhD from the University of Minnesota, USA. Dr. Dash is the professor and chair of the Department of Pharmacy Sciences, Creighton University, and the holder of Gilbert F. Cafe Jr. Endowed Chair of Pharmacy. He serves as the Associate Dean for the research for the School of Pharmacy and Health Professionals. He has made important research contributions in dosage forms, phase identification, phase quantification in solid matrices, and development and evaluation of novel drug delivery systems. Dr. Das serves as on many journals editorial boards and is a fellow of American Association of Pharmaceutical Sciences, AAPS. With this short profile, I welcome cordially Dr. Dash to deliver his first lecture on this morning in the second session of one-day virtual 12th Indo-US Conference of Association of Pharmaceutical. I welcome Dash. Dr. Dash, over to you.
Thank you. Can you hear me and can you see me?
Yes, sir, you are audible.
Yes, sir.
Okay. Can you see my screen? That's the
No, sir. I
Sir,
request
we are allowing to share the screen. You please share the screen. It will be allowed.
okay. Because I need my slides.
Sir, I,
Hello.
Yes, sir.
No, I cannot see my screen here. I just want to share
my screen.
Sir, we have allowed you to share your screen.
Yes, but
Okay, and you can share that screen.
I am trying my here, but I cannot say this is my screen. Okay, I'm stop scaring now. Now I'll try one more time. Share
Yes, sir.
screen.
Yes. Yes, sir.
Share your entire screen. For some reason, I cannot share my screen. Um, is Dr. Dahia there? I sent my slides to him. Can he share from the other side?
Sir, actually, the yes sir is not here. Please, you can
Sir,
try
I'm
one
here to
more
help
time.
you. Can
Sir,
Yes.
sir, your means your ceiling is viewable in this video. So, can you adjust it to your face and then after you can share your screen?
I will take the video. I mean, stops. Okay. No, for some reason I cannot share my screen.
in the private chat, I have given you the email ID. If you can share it, I will try to
Okay.
present it.
Let me see.
Have you got it in the
No,
private
not yet.
chat?
Okay.
In private chat.
Okay. I'll send that things to you. Okay.
Yeah, yeah.
I'm sorry.
No problem.
So did you get it?
Yeah. Sir, please tilt your camera so that it will show your face.
Can you see me?
Yes, sir. Yes, sir. We can see you now. Sir, we are going to share the presentation.
Okay.
And you can, then after you can
Okay.
continue. Sir, just wait for a few seconds. We are sharing it.
No,
Yeah,
no
yeah, yeah.
problem.
You got it?
Yes, sir, we have it.
Okay. Finally, we got it.
Yes, sir. Now
Can,
you can continue.
can you hear me now?
Yes, sir. Yes, sir. You are audible.
Okay. Thank you very much for the nice introduction, and sorry for all this inconvenience.
No
And
problem, sir.
so the title of my seminar today, or my talk, will be specially on decarbazine and resveratrol. Again, I'm missing my slides. Okay. And this is a preparation in lipid-based nanocarriers for the treatment of cutaneous melanoma. Uh, as the name implies, as you can see, this is a treatment options for cutaneous melanoma. Let me talk about, and we are talking about two different drugs which are we are planning to introduce to treat this disease called cutaneous melanoma. Let's go to the next slide. When we'll go to the next slide talks about, can I go to the next slide, please? Uh, can I change the slides from here? Possibly.
No, sir, you cannot.
Okay. So let's talk about skin cancer. What is a skin cancer? Because we are talking melanoma. It is the most common cancer in the United States, and it is the actually near about 10,000 people are diagnosed with skin cancer every day in the United States. So people, to understand how this cancer works, if you'll go to the skin, please go to the second slide, please. I'm still in the second slide. So as you can see, skin is made up of many layers. I'm just putting four, actually it is more than four layers. But as you can see, this depending and also many different types of cells. So to understand this particular disease, we have to understand a little bit the anatomy and physiology of skin. And as you can see that different in a normal epidermis, actually the major two layers are epidermis and dermis. As you can see in the epidermis, there are different types of cells. And some cells are called squamous cells, basal cells, and melanocytes. Depending on the cells, if this is impacted or the cancer impact those cells, it can lead to squamous cell carcinoma. It can lead to basal cell carcinoma, and it will be leading to called melanoma. That's the one we are talking about, which is the deadliest form of this cancer. And next slide, please. If we'll go back to the next slide, you will see that melanoma is a type of skin cancer that begins in a pigmented producing cell called melanocytes, as I showed you before. It is the most aggressive and the devastated form of the skin cancer, and actually 90% of the death happens because of this melanoma. And it predominantly occurs on the skin, that is the cutaneous, and it's called cutaneous melanoma, 90% of the cases. But in some cases, you will be finding this melanoma can happen also in other tissues, like including eyes, and we call it uveal melanoma. And it can happen also in different mucous membranes of lining of the body. and those are called mucosal melanoma. Let's go to the next slide, which will talk the different stages of this disease. Next slide, please. So you can see in the different stages of melanoma, actually these stages are from 0 to 5, 0 to 4. Actually, there are different 5 stages. So stage 0 stage, as you can see, is that is the starting, which is on the surface of the epidermis. And again, it is a localized portion of this disease. In the stage 1, as you can, from stage 0 to stage 1, it will be just increasing a little bit more thicker. Still, it is localized. Stage 2, you can see that means this, in fact, the cancer is growing more and more and becomes deeper. And still it is at this particular stage, stage 2, still this cancer is localized. It has not been metastasized, or it has not grown to other tissues. But if you go to the stage 3, actually this is where the problem starts. At this stage, they can go to the lymph nodes, and it can infect other parts of the body, and it becomes metastasized. However, this is the last stage, the stage 4, which is the most devastated stage. At this stage, it will be going to the blood circulation, it is metastasized. And this is the most important cases where the death rate is near about 90%. Whereas in the stage 3 can be 60%, whereas in other 0, 1, and 2, the death rate possibly will be within 5%. So I'll go to the next slide, please. In the next slide, I'm talking about the facts and premise of this study, what I'm talking about, or I'm going to discuss today some of the work we are doing. So we know that worldwide incidence of cutaneous melanoma has been rising annually at a very rapid rate as compared to any other type of cancer. So, and we are utilizing 2 different drugs, which is called dicarbazine, which has been approved as a monotherapy in the treatment of malignant cutaneous melanoma. But the problem with this particular drug is selectivity and tumor targeting. And it has a problem with stability and dose-limiting systemic toxicity of this drug. Whereas resveratrol, as you know, that it's a natural polyphenol found in wine and other materials that we consume, that has shown to inhibit proliferations of cell apoptosis in melanoma cells. And it has also been shown co-administration of these 2 drugs gives a synergetic effect as well as reduces the toxicity. So the premise of this study is to design and characterize an injectable nanostructure lipid carrier, I will call it NLC, for targeted delivery of both agents for the treatment of cutaneous malignant melanoma. And the mode of treatment of this drug delivery system will be by IV injection, because main objective is targeting. Next slide, please. So the objective of this work, can somebody help, please? The next slide. The objective of this study was to design and characterize a nanostructural lipid carrier system for targeted delivery of dicarbazine and resveratrol in the treatment of cutaneous melanoma. Next slide, please. So that is our major objective. So in order to find out or design a drug delivery system, the most important things we have to design or develop a analytical method that will be simultaneously quantitating both the drugs in a very sensitive manner. So we developed the HPLC chromatographic method for simultaneous determination of both the drugs. Next slide, please. And the parameters of this analysis are provided in the next slide. I'm not going through. Next slide, please. So this is the HPLC method, and all the chromatographic conditions are provided here. I don't want to go through this. And the next slide, if we'll go to the next slide, it will show you that separation, which is a specific method, and within 2 minutes we are separating the dicarbazine from resveratrol in 3 minutes, and we have a very linear um, linearity in the assay procedure for both the drugs. Next slide, please. That will be giving you the validation parameters. That means the method has to be validated, and we can see it is linear and it is a linearity range was near about 2 to 200 micrograms per ml for both the drugs. And we have also shown the interday and intraday precision as well as accuracy of this method, and the RSD is around less than 10%, which is actually as specified by USP and IHC. Next slide, please. Next slide talks about how do you make this nano lipid carrier. This nanolipid carrier is a simple system where you will be dissolving these lipids as well as the with solid lipid as well as the liquid lipid in a organic phase, and you will be adding that dropwise in step 2 to the oil phase in presence of a surfactants, and you will be forming an oil in water emulsions. And after this primary emulsion is done, the most important part is, because you have to make nanostructure, we have to apply energy, that is by a probe sonicator, and you will be getting the nano structure here. And these will be now this nanostructure, which will be just like a colloidal dispersion type of materials or dispersion, will be now freeze dried to give a free-flowing nanoparticles. Okay, so that means the nano carrier can be present in a liquid formulation, or it can be lyophilized or freeze dried to give a free-flowing powder. Next slide, please. This is some of those parameters, the characterization parameters we have done. I think again, we can see that in presence, the entrapment efficiency and percent of drug loading of these materials varies depending on the nature of the drug. DTIC, which is a dicarbazine and a resveratrol, you can see these two drugs are having different hydrophilicity and aqueous solubility and partition coefficient. So you'll be finding that both the drugs, dicarbazine has a lower entrapment efficiency and drug loading as compared to resveratrol. Next slide, please. And another important parameter when you design a drug delivery system, the most important things to find out is the physical state of the drug. We put two drugs in this lipid carrier. What is the fate of this drug? Is this drug present in a crystalline state, or it is in the dissolved state, or in the amorphous state? Why it is important? Because that will decide the release characteristic of the drug when will be administered these drugs to the human systems. So therefore, we determine the physical state of the drug by DSC, differential scanning calorimetry, as well as powder diffraction or powder X-ray diffraction. The next slide talks about the powder diffraction studies, and these studies has indicated that drug is present mostly in a non-crystalline state. That means in the dissolved state in those nano carriers. The next slide, please. The next slide will be talking about the characterization again. And in this particular characterization, we are talking about, we are having different types of nano lipid systems. Some are blank, some are drug-loaded nanoparticles containing both drugs, and also you will see pegylated nano structure. The region of this pegylation is, as you know, this nanostructure when given IV, it will be possibly eliminated from the body very quickly. But our objective for targeting, we want to keep those nanostructure for longer time in the circulation. Therefore, by pegylation, by putting a PEG on the surface, we call surface modification, you can keep those particles for longer time in the systemic circulation, and you can target to the tumor cells because of their nanostructure, because of their properties, as you know, that extra permeability and retention properties. Go to the next slide, please. This is also another study we did, because as you know that I'm talking about that this nanostructure was lyophilized or freeze-dried. During this freeze-drying procedure, we can see the presence of this freeze-drying. That means when you do freeze-drying of the structure, we think the freeze-dried product is becoming larger particles. So even though we have very nano small particles, smaller particles in the non-freeze-dried emulsions, when these are freeze-dried, because of these agglomerations, we are finding that these particles in a freeze-dried product is becoming larger in size. But our objective is to make this size smaller so that we can easily target. Therefore, we utilize some cryoprotectant. Next slide, please. We used 3 different types of cryoprotectant to protect so that the formation of this agglomeration will be minimized. And we have used sucrose, mannitol, as well as trehalose, and we found 2% trehalose gives the better freeze-dried nanoparticles with the right size for targeting. Go to the next slide, please. And that is also confirmed with a scanning electron microscopy. As you can see, these nanostructures are spherical in sizes. Go to the next slide, please. So this is an in vitro release of the drugs from the pegylated as well as non-pegylated, both the drugs. As you can see, the drug dicarbazine is actually releasing very quickly as compared to a slow release of resveratrol. And that is because of the nature of these drugs. As you know, these drugs are with a different solubility as well as a different partition coefficient. Next slide, please. We also studied the toxicity of these materials when it is given to different cell lines. We use 2 different cell lines, B16F10 cell line, which is a cancer cell line, and also HECA cells, which are normal cells. And we compare the toxicity of these particular drug delivery systems with the drugs, without drugs, to see that whether this material are toxic or not toxic. Next slide, please. Since these materials supposed to be injected, we also carried out the hemolytic effect of these materials. We want to find out whether these materials are doing hemolysis, and we found these materials with the concentration at different concentration we are utilizing, the hemolytic effect of these drugs are very minimal. Next slide, please. And this is also a percent cell viability of excipients, because as you know that we are using different excipients for making these particular nanocarriers, especially stearic acid and also other oils, fatty oils. And we have found that stearic acid used in this formulation is becoming more toxic as compared to even the drug. So this particular excipient needs to be changed in this formulation because stearic acid itself causing lots of toxicity to the cell lines. Next slide, please. So in conclusion, I have to go very quickly to keep my timing. And so in conclusion, I can tell that robust and a sensitive HPLC method was developed, which can determine simultaneously both the drugs with very high sensitivity. NLC were successfully prepared and characterized for co-delivery of both the drugs and were in the size range of 200 nanometers, which are required for targeting when it be given intravenously. Both these drugs are present in non-crystalline state in the lipid matrix carrier and are stable over a period of 60 days. Entrapment of a resveratrol was seen to be higher as compared to the DTIC, dicarbazine, in the lipid matrix, and showed an immediate, whereas this dicarbazine showed an immediate release as compared to resveratrol, which will be given a slow release over a prolonged period of time. Next slide, please. The cytotoxicity studies revealed that the blank NLC exhibited considerable toxicity in comparison to even the drug-loaded NLC, and which is a red flag and needs further clarification, and also that these two substances we are utilizing them found to be more toxic, which are glycerin monostearate and stearic acid, and these two solids are need to be changed in this formulation. Hemolytic acids confirm that the presence of glycerin monostearate and stearic acid has no issues, no problem. It doesn't cause us that much of hemolysis if it's given intravenously, but they are very toxic to the cells. Future studies for this drug delivery system involves testing its efficacy and uptake in various cell lines. The possible synergetic of both the drugs in the formulation needs to be further clarifications, and also the modification of some of the solids. Here we have lipids, we are talking solid lipids, and needs to be changed in the formulation because of their toxicity. Next slide, please. Again, I want to thank Daniel Mont, my graduate student, and University of Minnesota for the XRD studies. Next slide, please. And thank you all. I'm sorry for all this inconvenience that I cannot present these things myself. Somebody has to do it from distance. And thank you all.
Thank you very much, sir.
Thank you, sir.
Hopefully, I was in time.
For everybody. So, yes, sir, you are in time, and our session is running in time. We are now moving into the next speaker, Dr. Sushama Talgaonkar. He is associate professor
For next session, I request Professor M. Vijay Anandi to introduce Dr. Sushma Talgaonkar, ma'am.
Yeah, thank you, ma'am. Good morning to Anna.
Good morning, ma'am. Good morning.
Good morning. Good morning. It's my pleasure to introduce Dr. Sushma Talgaonkar, Associate Professor, Department of Pharmaceutics, School of Pharmaceutical Sciences, DPSRU, New Delhi. Dr. Sushma Talgaonkar is working as an Associate Professor in the Department of Pharmaceutics, School of Pharmaceutical Sciences, DPSRU, Delhi Pharmaceutical Sciences and Research University in the Department of Pharmaceutics. She has about 21 years experience in teaching and research. Dr. Talgaonkar has granted recently 1 Indian patent and filed 6 Indian patents. She has published more than 220 research papers in high impact factor, international reputed journals, and 30 papers in national journals with H-index 46. It's really a great, amazing thing, madam. And I-10 index is 120 and 8,550 Google Scholar citations. Wow, it's really great, madam. Besides this, she has co-authored 4 books and authored 12 chapters in international reference books. She has presented papers in more than 50 national and international conferences and participated in more than 50 symposia and workshops. She has traveled to Canada, United Kingdom, and Singapore to present her research papers. Recently, she has conferred Professor C.J. Shishu Award for the Best Research in Pharmaceutical Sciences by APTI. She has also been conferred the UGC Research Award 2014-16. She has also received Best Paper Award in Pharmaceutics for article published in IJPR by APTI in its 18th Annual National Convention 2013. Her research paper titled Development and Bioavailability Assessment of Rimipril Nano Emulsion Formulation has received Most Cited Research Paper Award on behalf of Elsewhere and Board of European Journal of Pharmaceutics and Biopharmaceutics. She has awarded the prestigious Motan Devi Dandia Biennial Prize for Best Publication in Pharmaceutical Sciences for the period 2010 to 11. Dr. Talgaonkar has so far supervised 26 PhD and 50 master's students. Currently, she is supervising 5 PhD scholars and PG project works of 4 students. She is actively involved in developing wide variety of smart and functionalized nano drug delivery system for targeted anti-cancer drug delivery. She has executed research projects sponsored by various national funding agencies like University Grant Commission, All India Council for Technical Education, and Department of Biotechnology. With a short profile and amazing profile, I welcome the Dr. Sushma Thalagonkar to proceed our talk. Thank you, madam. Thank you, all the team members.
Okay, thank you so much for wonderful introduction. Is it visible? Hello?
Yes,
Yeah,
ma'am, you
madam,
are
visible.
visible.
Okay, great. Thank you so much for wonderful introduction. Before starting this my presentation, I am very much thankful to Professor Dahiya, and who is the founder and the president of APP. And I am having a very great association with Dr. Dahiya. I am again thankful to Professor Sunita Dahiya, who is General Secretary, and I am associated with her since long time. Another the important thing I would like to thank to Gujarat APP branch, who has organized this wonderful conference, and a very wonderful talk is given by the speaker. And I am very thankful that organizing committee of Gujarat State Branch has given me this opportunity to share my some research work with all the great participants. So I welcome you all the students and the dear participant, dear colleague to this particular Indo-US conference. Today I am delivering a talk on the functionalized nanoliposome for effective treatment to rheumatoid arthritis. This particular talk, I will, the content of my presentation, although I short this presentation as due to the constraint of time. So outline the presentation introduction, objectives of my study, rationale, why I have taken this liposomal preparation for rheumatoid arthritis, and what was the hypothesis behind that, and formulation development of this nanoliposomes, characterization, conclusion, and the references. So the first slide is introduction. As everybody knows, the rheumatoid arthritis is a very challenging and autoimmune disorders, and we generally
Sorry,
define
ma'am,
as a.
for disturbing you in between, but your screen is not been shared still.
It is not shared.
शेयर
No.
करे तो यहाँ पे एक्सेप्ट करेंगे सर.
Please share. You will be trying to share. We will be accepting, and then after it will be visible.
But I have shared actually.
सभी वही पर क्यों हो रहा है?
Sir,
नितेश भाई सभी वही बोल रहे हैं, ये भी नहीं हो रहा है.
have you shared your screen, ma'am?
Share
There is a symbol of share screen.
Yeah. I have done, and I have allowed also.
Allowed.
I think there is some issues with that one.
I have allowed.
Screen, you just click the screen. Have you shared the entire screen, and then after you
No,
click the
no. When I'm sharing, then the share screen option is coming.
Yes, ma'am.
Now
Entire
your
screen. Share the entire screen.
now the your browser has blocked your screen. Click the screen
No.
icon in the your address bar to allow the screen.
Browser has blocked.
Ma'am, can you please leave, matlab stop sharing, and then after, can you please try once more?
Okay. Just a minute.
अरे हमें स्क्रीन शेयर करो, हमें स्क्रीन शेयर करो एक हमें क्लिक करो ना.
Hello, ma'am. Your screen is been able now. You can share it now.
Now it is screen. Entire screen करना है? Yes. Is it visible now?
Yes,
Hello?
ma'am. Yes, ma'am. It is visible now. It is visible
Okay.
now, ma'am.
Okay. And am I audible?
Yes, ma'am. You
Yeah, yeah.
are.
Okay. Sure, sure. So previously, what I have told, nobody has listened actually, or my voice was coming
Your
previously.
voice was clear.
Okay,
Your
okay.
voice
Then
was
it is
clear.
fine, fine, fine. So this is the outline of my presentation, and this is the first slide, which I am background I am giving for this rheumatoid arthritis. So everybody knows the rheumatoid arthritis. It is an autoimmune disorder, and till date nobody has found the reason why this rheumatoid arthritis is going to be happen in the maybe in the young generation also, and maybe in the old people. So there is no demarcation that this particular disease is going to be occur in only the aged people, but it can be occur at any stage. So it is an autoimmune disorders, and there are so many reasons which I will discuss later. And mostly it is ultimately, if it's not treated, it will ultimately lead to the deformity and the disability. So it is also a socio economical concern also for this particular disease, and it affects around 0.8% of the world and about 1% of the Indian population. It has also been found that the women are more vulnerable, 3 times more as compared to men, maybe due to the hormonal changes, and the other reasons are also has been researched. Now, the causes of rheumatoid arthritis, the slide is showing everybody, I think it must be knowing also. The mainly it is largely unknown. The people are the doctors and physicians, they are giving the permutation and the combination of the various treatments. The theory behind this particular rheumatoid arthritis is that it may be due to the genetics also, and maybe due to the environmental factor also. But possible triggers for this particular disease, maybe it has been reported, maybe due to the infection, gut and gut bacteria. So it may be the disturbances in the gut bacteria, maybe due to the smoking, maybe due to the obesity, and maybe due to the stress. Stress is a very one of the most important cause for the various autoimmune disorders. So one should try to make a stress-free life. And the female hormone is another major concern for why it is more in the female population. These are the some right side. I am showing some treatment modalities for rheumatoid arthritis. So basically, if you will stop the smoking, if you will do the physical exercises, if you will make your life very stress free, so definitely you can prevent yourself, because these are all the treatment as a prophylactically has been found out by the some researchers, and it has been reported also in the Therapeutic and various pharmaceutical magazines and all. Now let us see what is the pathophysiology of the this rheumatoid arthritis. So everybody knows whosoever is suffering from the arthritis, although there are so many types of the arthritis, rheumatoid, osteoarthritis, juvenile arthritis, and so on. But here I am deliberating on the rheumatoid arthritis. So especially the person who is suffering from the rheumatoid arthritis, basically he or she is having a pain, inflammation, and we can say the pain and inflammation in the joints. And there is especially for the penis formation in especially is near to the joint. And it is due to this inflammation, the tissues are going to be narrowed down, and there is no space for the movement and all. So it is going to be stiff and stiffer. And the person who is so suffering from the arthritis, it is finding a little bit difficult for movement and day-to-day work. And especially I have I have told that this is a particularly autoimmune disorder. So body is making the antibodies against its own mechanism. So it is also known as an auto antibodies and inflammatory cytokines. These two things are triggered especially in the human body. And that person is very immunocompromised, means their immunity of that person is very low, and he can very prone towards the other various infections also, because his body's immunity is compromised due to the preparation of the antibodies against his own antibody. So that's why the disease is very challengeable, and the people are very finding it very difficult taking the different medication, and a lot of side effects they are generally suffering. This is the slide which is showing the treatment modalities nowadays. So first line treatment, whosoever is going to the physician, they are, especially when we are saying ki I am having a pain in the joint, so they generally give you the oral and the topical therapy. Some ointments like the this diclofenac sodium and the gel, different type of the topical gel, they generally say ki you apply. And orally treatment are the non-steroidal anti-inflammatory agents and corticosteroids. Non-steroidal, as we all know, ibuprofen, diclofenac, they are very common. And corticosteroids, prednisolone is very, if the pain is very persistent and very acute, so definitely they can switch over to the steroids, which is not very advisable to take. But if you are not, the patient is not feeling comfortable, so a doctor has to prescribe the steroids. Then the second line treatment, the disease modifying anti-rheumatoid drugs, they are nowadays currently the first line treatment due to the severe side effects of this first line treatment. So disease modifying, they are again divided into the two, non-biological and the biological. Non-biological, they are the methotrexate, leflunomide, sulfasalazine and hydroxychloroquine. And biologicals, various antibodies, they are in the market, but they are very difficult to approach to the normal people because due to their cost constraint, and they are not suitable for each and every patient. So a very thorough investigation of that patient is required, ki what type of the antigen is there or what type of the antibodies the person is, that patient is having in the body. So these are all the antibodies, but they are not very, we can say, the common for all type of the patient. It is generally a one-to-one interaction of that antibody. So maybe infliximab is maybe suitable for one person, but it is not suitable for the some other person who is suffering from the antibody. So what I mean to say, the person has to go for a various biological investigation and various tests, and then he can find out which antibody will be useful for this type of the rheumatoid arthritis. And the third line treatment is that if the person is still finding after treating with the first line treatment, second line treatment, but still is not feeling or she is not feeling comfortable, then definitely he or she has go for the synovectomy. It is a surgical procedure where we can remove the synovial fluid and the particular joint or replacement we can do. So this is the treatment modalities. Now the next is the rationale of the study. Because the functionalized nanoliposome here, what is the rationale? I have taken here the disease modifying anti-rheumatoid drug leflunomide. So leflunomide's the active metabolite is the teriflunomide, which I have taken this particular project. So teriflunomide is again it is we can say the disease modified anti-rheumatoid drug. Because as we already seen in the previous slide, all the NSAIDs and the corticosteroid, they are having a lot of side effects. They are generally making a stomach upset, a gastric ulcer also. The person is not feeling very comfortable. So DMRD is the answer, and here I have taken the teriflunomide. The teriflunomide, if you will see, it is a we can say it is generally leading to the this particularly teriflunomide is mitochondrial. It is generally inhibiting the dihydroorate dehydrogenase enzyme. This particular enzyme, which is responsible for pyrimidine synthesis, and it is generally useful for proliferation of T and the beta cell. Now again, I want to clear here, the person who is suffering from the rheumatoid arthritis, they are having a very high level of this T cell and the B cell. This T cell and B cells, they are the we can say they are the immunological cells, and especially in the rheumatoid arthritis, their level has been increased. And this T cell and the B cells, they generally release the cytokines, which is a major cause found in especially rheumatoid arthritis. So all the intraleukin and the cytokines, their level is very high because the T cell and B cell levels is going to be increased. But here the teriflunomide, it is generally stop or it will inhibit this particular enzyme dihydroorate dehydrogenase. So the level of this T cell and B cells, they are going down, and the body is not producing the interleukins, IL-1, IL-6 and the cytokines, and the patient can feel very comfortable by using this teriflunomide, or we can say the disease modifying anti-rheumatoid drug. Second thing which I have taken here in this particular project, nanoliposome. As we already know, the liposome is the first preparation, controlled release or novel drug delivery system, which is a commercially viable. And till date, I think very few preparation of nanoparticle, but the liposome is the preparation which can be explored everywhere, whether it is a drug delivery system, whether it is a cosmetic technology, whether it is a agriculture, whether it is a food industry. These liposomes, they are the beauty of this liposome is that we are having a both compartment, lipid and the aqueous compartment. So we can entrap the two drugs very easily. Even one of my student has worked by taking this liposome specially for the melanoma treatment, and we found a very good result. We filed a patent also. So here also in the liposome, the thing is that the teriflunomide is a lipophilic, and liposome why we have taken, because it is a biocompatible, biodegradable. A lot of phospholipids are available for the selection also, and flexibility to couple with the site specific ligand to achieve the active targeting, because the phospholipids, they are having both the groups, hydrophilic and the hydrophobic groups. So we can easily put the ligands on the surface of the liposome, and they are prepared, and their scalability is also having no issue, because the method which is generally used for their production is very simple and easily available, and they are not very costly. So this is the reason why I have taken the liposome for this particular project. And the third thing which I have taken for this particular project to make this functionalized nanoliposome, it is a chondroitin sulfate. Chondroitin sulfate is a important ligand for the CD44 receptor. CD44 receptors, they are overexpressed on these T cells and the beta cells. They are especially the immunogenic cells, which levels has been increased in the arthritic patient. So they are overexpressed on the arthritic patient. So if I am taking the chondroitin sulfate on the outer surface of the liposome, so definitely the chances are that that liposome is going to bind with those cells which are having a CD4 receptors. And after binding that particular CD4 receptor, that cells, they are not producing the interleukins. So patient is feeling very comfortable. And another important parameter of this chondroitin sulfate is that it is a natural carbohydrate polymer which is present in our body, especially in the ligament, bone, and the joints. So if we are coating this liposome with the chondroitin sulfate, so definitely the formulation is going to the target area, especially joints, and in that joint it will remain for a longer time. Chondroitin sulfate will be there. So at the same time, they will release the medicament teriflunomide to the particular site where the action is desired. Along with the chondroitin sulfate is also going to be released, and it will cause the regeneration of the osteoblast, and articular regeneration is going to be happen. So along with the treatment, the regeneration is also going to be occur. So healing process is also going to be occur. So all these nanoliposomes, they are generally providing a 4 type of the action. First, the liposome is specially biodegradable, so there is no harm, no toxicity at all. Teriflunomide we are targeting because teriflunomide is causing a liver toxicity. So basic purpose of making this targeted system is that the patient should feel very comfortable, because teriflunomide, when the patient is taking in the form of tablet, the liver toxicity has been reported, and the patient is feeling very we can say the very discomfortable by taking oral. So here we are targeting the teriflunomide to the joint area, so the more efficient treatment for arthritis. Second thing, we are using the chondroitin sulfate. So chondroitin sulfate making as a ligand also, and at the same time it will remain in the joint side, so it is causing the articulate regeneration also. So healing process is also there, and it is also having a integral it is a extracellular matrix component, and also it will reduces the level of pro-inflammatory cytokines. So the whole is in if we see the system, it is acting as a synergistically. So we can also say it is the system is having a synergistic targeting ability as well as the active targeting ability. So it is a very robust system which can be used for treatment of the rheumatoid arthritis. Now the formulation development. The formulation development, I am not going in detail because the paper has also been published, but just to make it very clear, I have taken this lipoid S100 and cholesterol and chloroform methanol and hydration media as PBS, and this particular preparation is going to be prepared by the film hydration method, where we have used the rotary evaporator to make a thin film of the lipid. And in this organic phase, we have dissolved the teriflunomide, and after making a very thin film, we are hydrating that film with the phosphate buffer. And in order to get the small unilamellar vesicle in the nano size, we are going for the sonication also after preparation. After sonication, we have lyophilized the liposomal preparation. This is the whole steps it is given. And then after this preparation optimization we have done by using the QBD approach. And by QBD approach, we found the very optimized product, and they have given the 17 combination, and then we have optimized out of those 17 formulation on the basis of entrapment efficiency and the size, which are the 2 important parameter for the targeting of the drug to the particular area. So especially in the nano system, the entrapment is a big challenge. Rather, I should say the drug loading is an important challenge. for the formulator. And after this, we approach this optimization of formula, and here you can see the critical material attributes and the critical process parameter. These two things are very, very important for optimization of the product. And as you will see in this slide, I have taken the amount of drug and the amount of lipid and the volume of hydration media. All these three parameters, they are the critical material attributes, which is playing a very, very important role for optimization. And second attributes are the critical process parameter, means what instrument you have used, what is the steering speed, what is the temperature, what is the sonication speed, and the speed of the rotary evaporator where you are making the thin film. They are all the very important parameters to make a nanoliposome, which should their size should be below 200 nanometers, especially for the targeting. So after this, the particularly 2-3 formulations we have optimized, but the best result we have obtained from the formulation 10, where the size is 128.92 and its PDI is 0.21 and the entrapment efficiency is 73.94. So this paper we have published in the Drug Delivery Science and Technology for this in vitro characterization of this particular paper. Now, the second part of this this liposome is the functionalized liposome. How you are converting this, particularly the simple conventional liposome, to the ligand coated, that is a chondroitin sulfate formulation. So it is a, we have prepared by the ionic interaction. We have taken this particular mechanism. So first we have used the sterile amine to induce the charge on the liposome, positive charge, and then after that this particular liposome we have put into the chondroitin sulfate solution. So it is an ionic interaction between the sulfate and the sterile amine molecules, and due to this ionic interaction, the bond is going to be formed and the chondroitin is going to be adhere on the surface of the liposome. So we have optimized the chondroitin sulfate concentration also, and we found a very good result. The optimization has been done on the particle size and as well as the zeta potential. Sorry. So again, at the concentration, we have taken the different concentration of chondroitin sulfate 0.025, 0.05, and 0.075 and 0.1. We have taken this concentration from the literature review, and we have found a very good result at 0.05 concentration. So particle size is 155, and zeta potential is also positive, which I will show in the next slide for the targeting potential. Now the characterization of this without functionalized and with functionalized nanoliposome is shown in this particular slide. So the vesicle size is blank liposome. This is the teriflunomide liposome, and they are the this chondroitin sulfate liposome. So size is basically when we are applying the chondroitin sulfate coating, the size is little bit has been increased, but you will see the size has been found here in the 168 here in the chondroitin sulfate. And if you will see the zeta potential here, the zeta potential was in the minus, then it has been converted by putting the sterile amine positive, and then again it is going to be negative. So this is the again the characterization where we have performed the TEM and the SEM analysis. The TEM analysis, you will see the size is very well corroborated with the Malvern zeta size analyzer. And here this SEM and TEM study we have performed in the AIMS. So the size is very well corroborated with the particle size analysis. And this is the second slide is showing the chondroitin sulfate coating very clearly on the surface of the liposome. So these two figures are quite distinct, and they are showing the chondroitin sulfate has been adhered on the liposome. The second part is the same study, that is the surface morphology. And here you will see they are the small unilamellar nanoliposome, but if you are putting the chondroitin sulfate, some gel-like structure is going to be formed. Now this is the again important characteristic, the X-ray diffraction, because this liposome preparations we have lyophilized, so we generally see what is the nature of the drug. In my previous the previous lecture, the professor has shown this importance of this X-ray diffraction in order to see what is the teriflunomide XRD, what is the XRD of the chondroitin sulfate, what is the XRD of the teriflunomide loaded liposome, and what is the XRD of the chondroitin sulfate liposome. So we will see the lipid is showing, chondroitin sulfate is showing a particular peak at 20-degree angle, teriflunomide is showing the peaks on the 20 to 30-degree angle, while the teriflunomide liposome, they are showing the teriflunomide as well as the lipid peaks, and that shows the drug is going to be present in the not a very crystalline and not very amorphous form, and the drug is showing its proper entity, and we can say the drug is not going to be incompatible or not going to be interacted with the lipid molecules. Now, this is the again characterization of the formula, this formulation, because we are giving this particular preparation by the intravenous route. So the syringeability we have tested, hemocompatibility I have tested, and the osmotic fragility. These three parameters are very, very important because you are giving your preparation, you are administering your preparation by the intravenous route. So whenever we are giving this preparation to the directly to the blood, so we generally perform this particular all these three parameters. So first is the syringeability. We generally have taken the different gauge syringe needle 22, 23, 25, and 26, which is mostly used. And we have seen when we are our suspension of the nanoliposome, when we are injecting through this particular syringe, and then we find out the score. So number of the times we have injected through the syringe, and then we, after syringeability, we find out what is the particle size. Is there any difference in the size, or is there any difference in the entrapment efficiency? Because we are pushing when we are administering the liposome to the rat model or to the human being. So definitely this due to the pressure, maybe the liposome is going to be burst and the total drug is going to be released. So in order to making it very sure that liposome is generally very robust and they are not going to be disturbed due to the syringeability, that we have performed this particular experiment. Another important parameter is the hemolytic activity. What we have done, our mainly the this teriflunomide loaded liposome and chondroitin sulfate coated liposome, teriflunomide solution. All these three preparations we have put into the contact with the RBC. So it is the process has been reported. What we generally have done, we have find out if we are putting this particular all these three formulation which we are using in our research study, whether they are making any adverse effect on RBC or not. So again, fortunately, the plain distilled water is a negative control. It is not showing, it is a negative control. So definitely we have find a hemolytic degradation, and the hemoglobin is going to be come out from RBC. So the graph is showing the distilled water is showing a very lot of degradation of the RBC, and the hemoglobin is coming out, and we have analyzed by the UV spectrophotometer. Like this phosphate buffer, which has not been shown, that is a positive control. The phosphate buffer saline is having the osmotic pressure is equivalent to the blood, so it is not caused any harm to the RBC. And similarly, the teriflunomide liposome and teriflunomide solution and the chondroitin sulfate solution, they have shown a very little hemolytic activity. So here is the distilled water is a negative control, and the phosphate buffer, which has not shown in the graph, but it is a positive control. So phosphate buffer is as like as our blood we usually take for the release studies and all. So it is not going to make any harm. So similarly, our preparations have also not shown any degradation of the RBCs, and there is no hemoglobin has been coming out in our this analysis. The next is the osmotic fragility. In this osmotic fragility, again, what we have done, because already we have prepared, we are preparing a targeted preparation of the liposome. So definitely the liposome, when we are administering, it has to pass through from the site of administration to the site of destination. So our preparation should be very robust, and that's why we have checked what is the osmotic fragility, because the blood is having a osmotic pressure which is equivalent to the 0.9% NaCl. So we generally find out. So when we are administering this liposome, again we have find out, we have put our preparation with the different osmotic concentration. So hypo and the hyper. And we have find out whether there is any change in the size, whether there is a change in the number. We have taken this particular experiment we have done in the that hemocytometer, we generally do in the HAP practical, where we can count the WBC and RBC. So the same experiment we have performed in the with the liposome. By putting that liposome in the different type of the osmotic fragility solution, 0.1, 0.2, 0.3, 0.9 and all. And we have seen whether they are stable in the 0.9% NaCl solution or not. So we have find a very good osmotic, they have not osmotic fragile at 0.9% solution. So it is again sure that the preparation is very comfortable, and it is not going to be degrade when we are administering into the human body. This is an in vitro release graph where we have taken the three formulation, teriflunomide plain solution, then the teriflunomide loaded nanoliposomal preparation, and the chondroitin sulfate. coated nanoliposome preparation. So graph is showing a very sustained release preparation, although the reading has been shown for 24 hours, but we have performed for 72 hours, but I have not shown in this particular graph. But further, my student has worked for 72 hours also, and we got a very good result. So in this graph, we can easily see the drug is showing the effect only up to the 8 hours, while both the liposomes, they are showing a very sustained release pattern, 73.96% in 24 hours and 85.33% in the in the 73.96, and the teriflunomide liposome has shown the 85.33, while the drug is showing the 93.42 in the 8 hours. So that formulation is showing a very sustained release pattern of the drug. Now, another important study which we have performed in order to prove whether your formulation is binding to the target site or not, we have performed the in vitro cell line studies where we have shown that CD44 receptors, the chondroitin sulfate, when we are coating on the liposome, that liposomes, they are going to bind to the CD44 receptors which are overexpressed on the macrophage cell lines U937s and the MG63. So 3 cell lines we have taken in order to prove our targeting ability of our functionalized nanoliposome. So raw 264.7 murine macrophage cell lines I have taken. U937, they are the human macrophage cell line. In order to prove that the system is working in both the means in the rats model also and the human model also. And the MG63, that is the osteoblast, in order to prove that chondroitin sulfate is having a osteogenesis property. So the 3 cell lines we have taken and we have performed the experiments. Now, this cytotoxicity raw 264.7 cell lines, we have performed the study. If you will see the diagram, in the diagram, the chondroitin sulfate liposome, they are showing the maximum cytotoxicity. So what it is indicating that macrophage, and here again I have taken the liposaccharide stimulated and the liposaccharide non-stimulated. This lipopolysaccharide stimulating cells, why we have taken? Because if we are treating the raw cell, that is called the macrophage cell lines, and macrophage cell lines level is generally increased in the rheumatoid arthritis. So that's why we have taken this particular cell line. These 2 cell lines we have taken unstimulated and stimulated. But because when we are treating the cells with the lipopolysaccharide, the cells are showing the CD44 receptors. And that is a type of model which is we generally follow in the rheumatoid arthritis. When we are not treating these cells with the lipopolysaccharide, we are finding the result in a very different manner. So these 2 cell lines which we have taken, and this graph is showing in the raw 264 murine macrophage cell line. These 2 graphs, they are showing very clearly. Here in the first graph, we can easily see the level of cytotoxicity is very high. So what does it mean? When this particular chondroitin sulfate liposome, when we are giving to the rats which are having a arthritic induced model, we are finding a very good result, because in the lipopolysaccharide stimulated cell line, the CD44s are overexpressed. So chondroitin sulfate coated nanoliposomes, they are having a very good binding affinity towards the cells, and they are very successful in killing the cells. So that's why the toxicity is very high. While in the another group of the cells which are not lipopolysaccharides stimulated, here the reading is very low as compared to this particular. So here is the reading about the 79. Here is this in the case is the near about the 60. So result here in the graph, which in the table we can easily see, the blank liposome is not applicable. Teriflunomide is showing the cytotoxicity IC50 value at 30.2 microgram per ml, while the same teriflunomide liposome, they are showing the IC50 at 2.6 microgram per ml, while the chondroitin sulfate liposome, they are showing 0.034 microgram per ml. So by this table, we can easily say where the plain drug is required 30.2 microgram, but if we are putting the same drug in the chondroitin sulfate liposome, the concentration which we required 100 times has been reduced, and we can easily say the first step that if this formulation is successful, the liver toxicity is not going to be occurred, and the dose has been reduced, and the patient will feel very comfortable. So this is the first step which we have proved the targeting ability of our formulation. Another important thing which we have performed in the U937 human macrophage cell line, in order to that particular system whether it will be working in human being or not. So this is again that we have performed, and again the same result we got. The chondroitin sulfate liposome, they have shown a very good binding capability towards the CD44 receptor. Here again the table IC50 value for the CS liposome, it is the 13.6, while the teriflunomide is 62.78 required, and teriflunomide liposome is required 31. So just half concentration of the drug will required when we are giving in the liposome which are coated by the chondroitin sulfate. Then the third cell line study, again to prove that chondroitin sulfate is showing osteogenesis activity or not. Here what it is totally contrast to the previous experiment. Here we have taken the osteoblast cell line. Osteoblast cell line, they are generally required for the genesis, or they are the good cell lines which we require in the rheumatoid arthritis. So whenever we are giving any medicine to the patient, that is again the same, because chondroitin sulfate hyaluronic injections, they are available in the market. So if the person is having a very severe arthritis and he has lost his cartilage and the bone, so chondroitin sulfate injections, they are generally given to the patient. So what we have done here, in order to prove our hypothesis, we have taken this osteoblast cell line also, and here is the contrast to that, we have found a very good result. In all cases, if you will see at this concentration 0.003 to 33 microgram per ml, we have find out a very good result in the chondroitin sulfate. And the graph is here. We have plotted between the viability, not the toxicity. That is a major difference between the macrophage cell line and the osteoblast cell line. Here we have taken the viability, how much cell growth has been increased. So we can say in all cases, the this yellow is chondroitin sulfate liposome. They have shown a very good the viability or the regeneration capability. And in another word, we can say the liposomes, they are having a very biocompatible nature, because in all the preparation we have shown both the blank teriflunomide loaded liposome and the chondroitin loaded liposome, they are not showing any toxicity, and in each case the viability has been increased. So it is again proving our chondroitin sulfate coating on the liposome. Now, the another important study which we have performed, that is the cellular uptake study, and here we have taken not the drug, but we have taken the rhodamine and the DAPI we have taken. DAPI has been used for the nucleus staining, and the rhodamine is a proper particular solution. So here we can say, am I audible? Am I audible?
Yes,
Hello?
yes, you are audible.
Okay,
Yeah,
okay,
ma'am,
thank you.
you are
So
audible.
here in the cell line study, which is clearly shown that when we are taking a plain rhodamine solution, there is no fluorescence we got, because plain rhodamine solution is not going to penetrate in the cell line. But in the similar case, when we are going for this, again we have taken the here in the lipopolysaccharide positive and the positive cells in order to prove our result. So here in case of CS coated liposome, we will see a very good fluorescence, because this chondroitin sulfate is showing a very good binding on the cell line. So uptake will be more as compared to the plain without coated chondroitin sulfate liposome. Third slide. So here the fluorescence is less as compared to this chondroitin sulfate coated liposome. That again shows when we are making the coating of chondroitin sulfate, the cell uptake will be more, and the more killing of the macrophage cell line. And patient if we go for a further characterization, it is showing a very positive result by putting the chondroitin sulfate in our preparation. Now this is a graph which is showing a cellular uptake, and particularly they are showing a very good uptake of the liposome. So this is the dye solution, this is the rhodamine liposome, and this is the chondroitin sulfate liposome. So that shows a very good scatter type of the uptake study, which shows the chondroitin sulfate will be very helpful for the intake of the liposome. Now, another important study which we have performed, that is a pharmacodynamic study, again to prove that the formulation is working in the preclinical trial. So here the pharmacodynamic study which we have performed, and here the arthritic has been induced in the rat model, and this CFA model, that is a Fruand adjuvant model CFA, has been used for the induction, and it is an antibody injection which has been administered into the rats. And here we have taken the 5 groups of the rats, and one is the normal control, which is not, in which we have not induced the arthritis. Other 4 groups, they have induced the arthritis by Fruand adjuvant model, which is one of the commonest model for the rheumatoid arthritis. And all these 4 models, they require the 2 weeks, at least 12 days to induce the arthritis. And after 12 days, that the 13 days, we have started our treatment. So the first groups has been treated with the simple PBS solution. while other 3 groups which have treated continuously with the teriflunomide solution. Another group of the rats which has been treated with the liposome entrapped with the teriflunomide, and the 5th group has been treated with the chondroitin sulfate coated liposome. And injection has been given by the intravenous route because we are making it for the active targeting. So for the targeting, we can go for this intravenous injection. Now, the study has been pharmacodynamic. There are so many parameters which we can evaluate, although I have taken only the few parameters for this presentation due to this time constraint. So arthritic assessment, here we have taken the joint, the arthritic joint diameter, and here we have second point is arthritic score, which we have calculated. That is a particular ratio between the normal control and the induced. So we have taken the diameter of the induced arthritis because the inflammation has been induced. So definitely the swelling is going to be occur, and while the treated group, the swelling has been reduced. So that is the ratio which we have taken. And another graph is showing that chondroitin sulfate liposome, they have shown a very much significant reduction.
Sorry, ma'am, to disturb you, but you have just 5 minutes to complete this.
Okay, okay,
Yeah.
okay. Thank you so much. So this is again second experiment we have performed. Then this is a radiographic analysis of the same rats which we have which we have taken the X-ray of the arthritic induced rats, and we have find out that in the AC this mean radiological score is very high, while the teriflunomide solution, they have reduced this radiological score, while teriflunomide loaded liposome, they have also reduced, but maximum reduction and the significant reduction in the arthritis score was found with the chondroitin sulfate liposome. So this is again study which we have performed to prove whether the chondroitin sulfate coated liposome, they are working on the arthritic model or not. Then the next parameter which we have studied for the characterization, the histopathological of the ankle joint. So here again we have seen, we have taken the joints from the arthritic induced after completing the treatment and the performing the studies of 1 month. We have taken the different joints and we have go for the histopathology. And all these 5 slides, they are showing that in the chondroitin sulfate coated, there is no vacuole, there is no cell reduction, no pennus formation, and no vacuoles we have seen. And that shows the chondroitin sulfate is going to perform the osteogenesis activity, while in the first slide, that is a normal control, which is generally normal without arthritic we can see, while in the this particular is the arthritic induced rat. This 3 is the teriflunomide coated, and this is the liposome which is containing the teriflunomide, and this is the chondroitin sulfate coated liposomal slide. So we can say this particularly when we are induce the arthritis, there are the synovial infiltration which we generally found, then the pennus tissue formation, that is the PT, and the teriflunomide solution treated group showed the inflammatory cells also, while they have been removed in the case of chondroitin sulfate coated liposomal formulation. So another important study which we have done, which is again very important because the beginning of this project or the conception of this project has been started, that in the rheumatoid arthritis, the particular pro-inflammatory cytokines level is generally very high. So we have taken this 3 TNF alpha, interleukin beta, and the IL-6. Although there are so many biomarkers are available for this rheumatoid arthritis, but we have taken the kit for this TNF alpha and the IL beta and IL-6. So this particular blood has been removed, and we can go for this ELISA reader. We have analyzed the level of all these 3 biomarkers, and we have found a very positive result that chondroitin sulfate liposome and the teriflunomide liposome, they have significantly reduced the level of cytokines TNF alpha and interleukin 6. Now the next parameter which we have studied, that is again the liver tissue histopathology, because again we have said that it is plain teriflunomide cause the liver toxicity. So again we have done the liver histopathology, and very good result we got that when we are coating the liposome by chondroitin sulfate, when we are putting the teriflunomide in the liposome, we can save the liver toxicity. So while the teriflunomide plain is causing the toxicity, which we can see in the slide number 3, and but in the 4 and 5 we have not find out any we can say the damage to the liver cells, and we can say when we are giving the teriflunomide by the functionalized liposome, we can reduce the toxicity by 60 to 70%, because the teriflunomide is not going any other part of the body and it is going only to the target area. So we can prevent the liver toxicity of this formulation. So this is a again biodistribution study. I am just summarizing quickly because the 5 minutes are left. This particular work we have done in the DRDO in mass lab, where we have tagged our formulation with the technetium, and in order to find out whether our formulation will be retained in the joint side or not. So again, arthritic model we have taken, and we generally take the technetium and on the liposomes, and very good result we got, and we have find out that maximum our drug has been retained. It has been performed in 1 hour, 6 hour, and 24 hour in gamma camera. So in 24 hours, if you will see, the maximum formulation has been found in the kidney, and still it has been found out in the joint also. While in the first hour, the formulation is taken up by the liver, and very high concentration in the liver we have find, but at the 24 hours, the concentration of our particular formulation, they are still find in the kidney, and the joint is in the paw, it has been find out. So we can say the formulation is working because it is a ROI, means the region of interest. Here we can see in the slide, the still in the 24 hours, we can say the it is showing radiation after 24 hours in the camera. So that is our purpose has been solved by using the chondroitin sulfate that will retain in the joint for a longer duration, while in the normal simple liposome, which are not coated with the chondroitin sulfate, we have not shown any activity after 24 hours. So this is a stability study. The liposomes, they are quite stable, and 25 degree they have shown the 380 days and value, and the formulation is quite stable, although the further stability studies and the other further characterizations are required for this particular process. So in the conclusion, we can say the chondroitin sulfate is playing a very, very important role for targeting the liposome to the region of interest, and they are they can be proved as a good carrier for effective treatment of the rheumatoid arthritis. So this is the acknowledgement, which is this particular project has been funded by the CSIR. We are very grateful for the Intas Pharmaceutical, who has provided the drug sample. I am very much thankful to the Delhi University and the DABUR for carrying out the cell line studies. Same and the same we have performed in the AIMS. So we are again very thankful to the facility. This is the review and the research paper which we have published from this particular work. And again, I am very thankful to my student, Asia, who has worked this particular work. And now she is working in the multinational company. So thank you so much for patient listening and giving me this opportunity. Thank you so much.
Thank you, ma'am. Now the session is open for question, answer, and discussion. Professor Govindasamy. Yes, sir, you can answer.
Yeah. Ma'am, I think this is the right time to conclude the session, because the next session will be starting by the speakers are waiting on the line, I think. So may I request the organizers to collect the questions from the audience in a separate mail ID and forward to the speakers. Both the speakers in the morning, they have delivered a great lecture. First one, Professor Dash, he has given a lecture on the drugs on melanoma and the formulation evaluation. Now, the Madam Dr. Sushama has given a very good lecture, and I like to conclude both the sessions so that in the online event, the next session speaker by 11 o'clock. This is 11 now. May I request all the organizers to collect all the questions and forward to the speakers so that it will be easy for them. With this, I thank the speakers, both Professor Dash and Professor Sushama. Thanks for your elaborative lectures.
Thank you so much.
Thank you very much. Over to you,
Thank
organizer.
you. Thank you.
Okay, Professor M. Vijay Anandi, ma'am, if you want to say anything. Okay, so we are concluding our first session here, and the questions for this session will be sent to the speakers on their email. Okay, thank you so much, Professor Alekha K. Dash, Dr. Sushma Telangankar, Professor Govinda Sami Jayabalan, and Professor M. Vijay Anandi for sparing your valuable time for us. Thank you very much.
Thank you. Thank you so much. Thank you.
Thank you very much. Thanks for the opportunity.
Okay.
And all the best for the remaining section.
Yes, thank you so much.
Thank you, sir.
Thank you. Thank you, APP Gujarat.
Now we can leave.
Yes, ma'am. Yes.
Yeah. Okay. Thank you
Shall
so
I leave
much.
the studio now?
Okay, sir. Fine. That's fine. We are starting our next session in few minutes. Okay.
Good morning.
Good morning, sir. Good morning.
Hello. Hello.
Hello. Yes, sir.
Yeah. Yeah. Am I audible?
Yes, sir. Yes, sir. You are.
Okay. Okay.
Okay. So we are going to start our second session, which will be delivered by Dr. Atindra D. Shukla, Dharamsi Desai University, Nadiad, Gujarat, and Dr. Palanasamy Selvamani, Anna University, BIT Campus, Tamil Nadu. To this chair this session, we have with us Professor Ajay Sharma and Professor Nehal J. Shah. Professor Ajay Sharma is currently working as Professor, Department of Pharmacognosy, School of Pharmaceutical Science, Delhi Pharmaceutical Science and Research University, New Delhi. Sir has more than 14 years of teaching and research experience, has published more than 60 research articles, and has presented more than 100 papers in different conferences, and has contributed a book and 2 book chapters. Sir has filed 1 Indian patent and member of various national and international bodies. I welcome you, sir. Professor
Thank you so much.
Professor Nehal J. Shah is currently working as principal in Dubai Patel College of Pharmacy and Research Center, Dharmach, Anand, Gujarat. Sir has more than 20 years of teaching and 2 years of industry experience, has published more than 35 research articles in various journals, and has presented more than 28 papers in national conference and contributed a book. Sir has received a grant of 75,000 from a body, and I welcome you, Nehal sir.
Thank you.
Now, for proceeding ahead, I request Professor Ajay Sharma to introduce Dr. Atindra D. Shukla to us. Over to you, Ajay sir.
Yeah. Good morning, everyone. Am I audible?
Yes.
Yes,
Yes.
sir. Yes.
Okay. So, good morning, everyone. First of all, I like to congratulate the organizer who has organized this particular event, and they have provided me this wonderful opportunity to connect with all of you. It's my pleasure to introduce Dr. Atinder Sukla. Dr. Atinder Sukla, he's having a very vast academic and research experience, more than 70 years. And out of 17 years, he has contributed 8 years in academic field. And currently he is the director in charge at Center for Surface Sciences and Nanotechnology, Dharam Singh Desai University, Nadiad, Gujarat. And in, since 2011, he worked as associate professor in the same organization. He has also served various organizations in different capacities, like scientist C ad hoc at CSMCRI Bhavnagar from 2010 to 2011, and he was the scientist fellow at same institute from 2007 to 2010. He also had a postdoctoral fellowship from 2006 to 2007 at University of Miami, Coral Gables, with Professor Angel in the Department of Chemistry, and again he had postdoctoral fellowship from 2003 to 2005 at Vietnam Institute of Science, Israel. Very reputed organization it is. And he has got PhD degree from Central Salt and Marine Chemical Research Institute in the year 2002, and BSc and MSc degree from South Gujarat University and Bhavnagar University, India. Currently, he is working in the synthesis and characterization of novel materials and compounds. He is also involved in the designing and synthesis materials for delivery for various surface modifications, nanofluids for enhanced oil recovery, nanocomposites and their applications. He has guided more than 12 MPharm and MTech students, and he is guiding 8 PhD students right now. He has published more than 14 research papers of national and international repute, and currently he has invited for 20 talks, and he has presented more than 40 papers in various national and international conferences, more than 900 citations, along with H index 19 he has got. He has published 3 patents. Out of these three, one Indian and two international patent he has got, and 2 patents are under process. So it is a very wonderful opportunity for everyone to have Dr. Atinder Shukla with us. So over to you, Dr. Atinder Shukla, sir.
Thank you, sir. Thank you, sir, for an elaborate introduction. Should I share my screen now?
Yes,
Yes,
sir.
sir.
Okay, okay. Okay, is it visible?
Yes,
You
sir.
have
Is the
to,
screen visible?
sir, you have to share the entire screen,
Okay.
and then after we will be accepting it, it will be visible.
Okay, okay, 1 minute. Application window, your entire screen. Okay,
Yes, sir. Yes, sir.
share. Okay.
Done. Bolo.
Okay, sir. Is
Now,
it visible now?
yeah, okay, okay. Thank you very much. As Dr. Sharma gave an elaborate introduction, all of you might have heard that I am an chemist, not a pharmacist. But the theme of our conference is entirely interdisciplinary. So thank you for the organizers and the host institution for inviting me, Dr. Rajiv Dahiya and his team, and Association of Pharmacy Professionals for this kind of event and invitation to researcher like me. Today's topic of my talk is the nanotheranostics, nanotechnology aided diagnostic and stimuli responsive targeted therapeutics. Students might feel that this is too many jargons of very heavy words, but this talk is actually aimed to simplify it for all of you. So let us begin. And since the lunch is approaching, let us begin with starter. So it is our custom to start with starter. So let's go very some simplistic ideas about nanotechnology and the scale and all. So most of you know that nanotechnology deals with the structures, or we can say features, sized between 1 to 100 nanometer. That is nano, in the order of scale, we can see 10 to the power minus 9 with respect to the meter size. So all these below that meter are 10 to the power minus sign, that milli, nano, angstrom, pico, femto, or you can go up above the meter. And so essentially this dimension, we are talking about the size in terms of length. And when a material contains at least one dimension, or it involves developing or modifying materials within that size in 1 to 100 nanometer, then it is called nanomaterial. So that is where nanotechnology is dealing with these type of materials. These are called nanomaterials. So as we have the theme that nanotechnology in pharmaceutical sciences sphere, but let's begin with that where nanotechnology contributes. So it contributes in various spheres, not only in a medicine or pharmacy or health area, but also in food and agriculture, aerospace, surveillance, defense, security, transportation, biotech. All of us are knowing now how these virus related vaccines and development are now taking place. And we are seeing that with the aid of latest technologies, including nanotechnology and other tools, we are seeing the development of vaccine and everything became so faster with respect to the previous attempts to make vaccines for different types of older diseases we have seen. So there the technological advantages of nanotechnology and the combination of other technologies we are already feeling. Mechanical engineering, robotics also it is contributing. In information technology, advanced materials, textiles, energy and environment, this field is receiving tremendous contribution from nanotechnology nowadays, particularly energy harvesting, storage, all those area. And environmental remediation, of course, CO2 sequestration and other kind of applications. So nanotechnology is a very versatile kind of concept or field. It is contributing into various spheres, but today we are going to be limited up to medicine, health or pharmacy area. So what are the nanomaterials normally in pharmacy with the perspective of scale we can see? So 1 nanometer to 1000 nanometer, or 1 nanometer to 1 micron scale is given here. And we can see that right from the dendrimers, the polymeric nanomaterials, then metal nanoparticles, nanocrystals, block polymer based micelles, polymeric nanoparticles, liposomes, and nanosuspensions. Nanosuspensions will have a kind of nano materials or nanoparticles suspended in a typical system. So it is a kind of formulation. It is not a kind of single entity. Normally it is a formulation. So nanosuspensions are of a very larger size considering the other components of the system. And with perspective to the biomolecules, the hemoglobin is around 6.5 nanometer, antibodies are around 12, the virus, coronavirus or influenza virus, they are in this range of 130 to 200 nanometer. So, or below 200 nanometers. So this is the size range we are dealing with. So nanomaterials are already smaller than the biological materials, or in that range. So they are compatible to each other in terms of size, like not very tiny, not very large. They are kind of in the range domain or size domain of these biomolecules. Another thing is that why nanomaterial, or what type of nanomaterials in pharmacy. So nanotechnology aided therapy is basically newer approach, but it is maturing very rapidly in since last decade or last couple of decades, because the salient desirability remains same, like whatever an ideal drug or a novel pharmaceutical entity it is desired of, the same demands or same requirements are of nanomaterials also, that improving efficacy, improving solubility, diversifying the dosage forms. So certain dosage forms, like we have seen that previously vaccines were delivered through injection. Now biotech covaxin is coming in an intranasal form. So diversifying dosage form also can be aided by nanomaterials in pharmacy. Controlled release, it is one of the most desirable thing. Mitigation of side effects, it is also very important. Greater patient compliance, it is always there. And then affordability is a key point, but at the moment, like in the nanotechnology aided therapeutics or dosage are not going to be extremely affordable compared to the classical or conventional available options, but it is maturing, that science is maturing slowly. Affordability will also be achieved. So after enough of starter, let's go to main course, because too much starter will not be very good for health. So let's go for main course. We start with some keywords, because as I said that jargons of words might confuse the students, especially because the seasoned researchers are knowing these things, and those who are into kind of interdisciplinary collaborations, they know this, what are these words, what do they mean. And so targeted drug delivery is one of the very important aspect, that this term defines delivery of drug at the site of action without harming or affecting other organs or tissue or cells. So that is desirable, not always 100% achievable, but it is the desire of the whomsoever is designing the drug, be it chemist or pharmacist or combination of a team. They wish that drug should be targeted. It is not, they are not designing it to target to I mean say cancer drug going into some other. So this is one of the main fundamental aspect or fundamental requirement for drug designing. Another is particularly for this topic, or what we are dealing with, nanotherapeutic, that is it should be stimuli responsive. That means that when drug delivery mechanism is designed such a way that certain external or internal stimuli or stimulus can trigger the drug or the carrier to release the its constituents to the system or wherever it is incorporated. And then I mean, it's like it is a kind of switch based, like you press the switch and you are releasing something. So stimuli acts as a kind of trigger or switch for release of certain API or the pharmaceutical ingredients or the desired materials which we want to deliver into the cells, the disease cells. So there are certain stimuli like pH, temperature, photoirradiation of a particular wavelength, or presence of certain biomolecules or redox potential variation at the site, and magnetic field also sometimes. So this type of stimuli can be created or can be induced to have a desired effect. And then theranostic, it is a kind of little bit a new word, and it is a hybrid word. It is not a kind of a classical English literature word, but it is scientists have made this word by combination of two words like therapeutics and diagnostics. So combination is theranostics. So nanoscale materials, when they are used, it is nanotheranostics. Otherwise also it is theranostics. So sorry. Yeah. So now after that we are entering for our journey, we need a vehicle. So let's see some example of vehicles or carriers, a nano carrier. This is just an example, a kind of schematic, not necessarily all the vehicles or carriers used for drug delivery in nanotheranostics should be like this, but this is a kind of schematic example how it will be designed. Like you can see the lipid bilayer based kind of say liposome type drug delivery agent. In that you have on the walls, you have external walls, we have targeting ligands, we have anti-fouling agents, because we need I mean it should not get biodegraded before it is reached to the target site. And the targeting ligand will ensure that it reaches to the target sites, and then it binds there. And then there will be certain I mean on their construction, or in these constituents there will be certain agents which will respond to the stimuli and open this entire carrier or entire encapsulation up. And then these diagnostic agents and therapeutic agents will be entering into the cell or entering into the disease site. And diagnostic agents will give the signal to the various diagnostic techniques to probe whether drug or your carrier has reached there or not. And then therapeutic agent will start the therapy. So this is you can see the inherently it is very smart system by design for advanced drug delivery, and where it is used. So obviously this type of sophisticated system needs to be used again a very threatening or sophisticated disease type cancer, because it is life threatening. Still, after so many years of research, cancer still remains one of the highest of humankind. So this type of advanced delivery system definitely should target this type of diseases. So, but not limited to that. But here we are taking cancer as one of the example, or one of the worthy example to discuss upon. And why it is used? Because that's supposed to be more effective, accurate, less intrusive, and lesser side effect. That is always whenever a newer approach is trying to replace a classical or conventional approach of drug delivery or drug therapy. These are the points, like it has to be more effective and accurate, less intrusive, lesser side effect, and affordable also. So this is why it is promising in cancer treatment, because this is an alternative. So it is really novel. Its potentials are still unexplored, not fully explored. We can say now it is being explored, but still not fully explored. And it is promising in. I mean, it's like nanotechnology has proven its promise in other fields. So why not to try into this medical health or pharmaceutical sciences field? So that is one of the argument why this is being pushed. This nanotechnology, and certain unique properties and features which nanomaterials have, that like we saw for these nanomaterials which are used in pharmacy, we have seen in previous slides. They show these certain unique properties and features could be very promising, or could give us a kind of desired results. And there are certain examples that they are already giving, but not all of these drugs are already in market, nanotechnology based, nanotheranostics based. But research is going on, it is slowly maturing, and it will start delivering the fruits very soon. So there and why it is desired, because there were certain limitations in the current or previous therapies based on macro medicine based, particularly when nano was not introduced, that microscopic residuals remain, functional damage to various organs happen, development of resistance in cells happening, particularly in case of cancers. So these drugs are failing repeatedly with overdose, even it is causing severe and long lasting side effects. So that is why people are moving towards, or probing at least, the nanotechnology or nanotheranostics field. And now we are moving into the design part, like how this is designed. So nanotheranostics involve the major challenge is design of appropriate system, because what we saw as a concept, or what we proposed as a concept, it looks very good, like it is doable, but when we start designing, it is a very complex system compared to the conventional drug delivery or drugs which we are using. So it involves lot of different fields contribution, lot of different researchers, a team need to be there, like supramolecular chemistry and self-assembly to design the carriers. When you use, say, for example, liposomes or polymeric carriers and all. So those supramolecular chemistry self-assembly aspect will play some role into designing those things. Then sensing and molecular recognition concepts will come, because what we are using, these diagnostic agents, and there are certain targeting ligands, will do some kind of molecular recognition to achieve the targeted delivery. And then sensing will be done by, say, these diagnostic agents. It will show you the imaging. So this is a combination of sensing and molecular recognition. Then chemical biology, molecular biology principles involved very heavily. I mean, action depends on those phenomenon, because that what you are targeting, how it is binding, whether we are using a peptide or a peptide receptor type of thing, antigen, all those things. So those things involve chemical biology, molecular biology, biotechnology, all these fields contribution. Then detection and diagnosis is a kind of, I mean, where it involves a kind of some analytical techniques, where you do the detection and diagnosis using various tools. Of course, sophisticated instruments will be used, but where we need to have a kind of analytical knowledge, that how to analyze this kind of complex system. And then drug release, the control over drug release and its pharmaceutical action, the pharmacodynamic, which we call. So all those things then come into picture when drug is released into this. So this is a very stepwise, very complicated system, and its design is, that's why it is little bit tricky and difficult, but it is possible. There layer by layer approach and other approaches, people have shown this. So basically this approach is therapeutics plus diagnostic. So what we do is application of nanotechnology in preparation of dual purpose nano carrier. Basically, what dual purpose? Therapeutics and diagnostic. So it needs to give therapy also and diagnosis also, and both needs to be processing in a simultaneous manner. The combined result, I mean, it will result into the improved disease management by reducing time, because you diagnosis separately, then you decide the course of therapy, then you administer the therapy. It reduces the time, reduce the risk, because early detection and early therapy will reduce the risk, and eventually the cost also will be reduced, because the time is reduced. So hospitalization, treatment, everything is reduced. I mean, it's expected to be reduced. So this is the approach, and suitable therapeutic approaches can be tailored for all diseases, including cancer. So now we are taking case study of cancer, but it is not limited to cancer. We can design the suitable therapeutic approaches for various diseases. Because in all diseases, the target mechanism, the drugs, everything will be changed. So accordingly, we can design the specific carriers for that. And now we can move into the challenges. Challenges are relatively common for majority of drugs. Similarly here also, we have similar challenges like prevention of drug from biological degradation. So when it is entering into the biological system like our body, or and at that time it should not get degraded very quickly by other enzymes or other bodily fluids. It should be stable enough to reach up to the targeted site. Effective targeting should be there, so it should not be wavered, like you design to target cancer cell and it ends up with cancer cell also and some other cells. So that wavered targeting can cause damages. Patient compliance should be there, so dosage form should be chosen such a way that patient compliance increases, cost effectiveness and product life expectancy. So these are almost the challenges for all type of therapies, not only for this nanotheranostics. These are common for all type of. So these are there also in this case also. It is not that those things are overcome already, but efforts are made to address these challenges. At the same time, the other salient features for cancer nanotheranostics is diagnosis can be done using quantum dots or fluoro probes or radioisotopes, and tumor targeted delivery can be achieved by tumor cell binding ligands or specific peptides which target the receptors or antigens. Those type of tumor targeted delivery can be achieved. Imaging by say confocal microscopy or tomography, so that will be part of diagnosis. And on-site drug release against stimuli. So stimuli could be different types, or maybe temperature change, because the tumor cells will have a high temperature. Then we can have a pH, all those type of things. So these are the diagnostic agents or techniques, and therapeutics agents and techniques. So we are looking, I mean, I'm not going to mention all of this, but these are the various diagnostic techniques and the agents used for that. And probably most easier one will be the optical imaging, where it is performed by confocal microscopy. And therapeutics techniques and agents are like chemotherapy, photodynamic therapy, radiation therapy. That remains, but once targeting is done, then these therapies will be less damaging to the other organs and other tissues or cells. So that is the one. And the stimuli is plural of stimulus or trigger, and drastic change in certain parameters cause the stimuli, maybe the and then it releases the probes or drug molecules from the carrier. The intrinsic features of cancer cells to influence the carrier behavior, it include the extracellular and intercellular pH changes, temperature changes, glutathione concentration, because the near the tumor cells, glutathione concentration when cancer is progressing, it is higher. So it acts as a redox mediator and can be exploited to be a kind of the targeting molecule or even the stimuli also. And irradiation of particular wavelength can act as a stimuli for a photoresponsive system. If the carrier has a photoresponsive system designed, then once it is reached to the site, we know that from the diagnostics, then we can give a kind of wavelength of certain certain irradiation with certain wavelength. If we give, then it will release the drug. Or so this type of nano carrier preparation, these steps are involved. Mainly core cell type nanoparticles are preferred, where the shell could be of polymers or liposomes, normally softer shells, micelles. And then core could be a solid template or mesoporous material or metallic nanoparticle in the core, with the drug probe or binding sites are also there. And hollow or micro nano capsules can be also used. Micro I'm saying because many times a hollow capsule could be of a larger size and internal components could be of nano size, so still it can be counted as a nano technology based system. But these hollow region can host the drug and probe molecules, and it can be created by say solid templating, and other methodologies are there for creating the hollow templated nano capsules. And drug loading and probe loading is drug loading and probe loading is also very important. Loading of anti-cancer drugs should be taken place, and fluorescent or MRI active or radioactive probe molecules also should be loaded. Then layer by layer assembly, coating of stimuli responsive cell, all those steps takes place. In that you have to have a thermoresponsive layer or pH responsive layer or even redox responsive layers, which will be responding to the stimuli there. So this is the kind of concept. What I'm not going into very much details of it. We will see some examples how it is. It is already in 2019. This paper in cancers shows that how this schematically this system could be. So we can see the ligand modified nanoparticles hosting the drug, and then it has a various kind of ligands on the its periphery. They can bind. They are biological ligands. They can bind to the targeted disease cells. So this is I mean, when injected into the body, this is how it is going to work. This, yeah, yeah. And this,
चलो ठीक है ठीक है ठीक
yeah, yeah, targeting and binding the cells is another example. This is a little bit more specific example with the names of the peptides or the antigens and receptors which are going to be involved in this kind of targeted binding, and then eventually will ensure the targeted drug delivery. So this is also a very recent paper where they have shown the the bilayered lipid containing liposome type carriers delivering the targeted drug or targeted delivery of the drug. So these are the concept which we plan to work on, because we collaborated with one of the French group, and we thought of applying for a Indo-French collaborative project, which in the first attempt did not work, but we designed our system. Our concept was ready. We thought of smart copolymer based nano capsule. One was the hollow nano capsule with solid templated synthesis we thought of, where calcium carbonate was used as a template nanoparticle, and then it will be leached out. So we end up with a hollow nano capsule. Or also we have designed a micellar based system, and in both the cases drug loading will take place, and then this micelle or this I mean drug loaded micelle or this nano capsule will target the tumor cell with on this stimuli such will nanocargo rupture will take place and drug release will take place. So this was the concept which we proposed, and then obviously in the proposal we used the specific examples of which kind of chemicals or the polymers we are going to use. Here I am not putting them, but this is the concept which I am just showing that how it would be working and targeting. So this tailor-made capsule approaches the cell wall and then enters into that, and then slowly it goes and gets ruptured, and the biomolecules or the drug molecules get delivered. At the same time, probe molecules are also delivered. Then once probe molecules are delivered, we can diagnose that how much concentration of the drug or how much concentration of the carrier achieved, whether it is sufficient or not. And then when trigger is released, then this therapy comes into action. So this is how the nanotheranostics should be operating in the cancer therapy. And these are these common references apart from those few schematic figures and diagrams. These are the common references which one can refer. And I would like to acknowledge my colleagues, Dr. Bhavesh Bharatia, who is now currently in UK pursuing his postdoctoral research, Dr. Sabrina and Dr. Benjamin from our French collaborating group. Miss Vidisha is now Dr. Vidisha, and Mr. Mukesh Sharma was part of team at that time. Now he's not with our group. And our guide, and because as I said that I am a chemist, so I need guidance from expert pharmacy people, and those are Dr. B.N. Suhagia, Dean of Department of Pharmacy in our university, and Dr. Tejal Soni, she is a senior professor there, and our founding director, Dr. Dinesh Osa. He's retired from the he's emeritus professor with Columbia and Florida University, and he has a vast experience in psycho science and drug delivery and other aspects of the um, targeted drug delivery. So this was our team, and I would like to thank all of them and thank all of you for patiently listening this talk on nanotheranostics based targeted drug delivery. Thank you all.
Thank you so much, sir. Okay, so now I request Professor Nehal J. Shah, sir, to introduce Dr. Palanisamy Selvamni for the next lecture.
Good afternoon. Am I audible, madam?
Yes, sir, you are audible.
Well, let me congratulate Dr. Dhiren, management and team of Dr. Dhiren for organizing such a wonderful 12th Indo-US conference, which has been organized by Sri Lalji Bhai Patel College of Pharmacy, Umrak, in association with APP, where it's my great pleasure to introduce Dr. Palaniswamy Selvamani, who is from BIT campus, Tamil Nadu, and he is associate professor, Department of Pharmaceutical Technology, Center for Excellence in Nanobia Translational Research. He is MPharm PhD from Jadavpur
Hello.
University. Yeah, am I audible?
Yeah.
Yes, sir. Yes, sir.
Am I audible?
Yes, sir, you are audible.
Am I audible?
Yes, sir, you are audible.
Yeah.
हेलो
Okay.
हाँ
But I am
मैम
receiving some disturbances.
मैडम मुझे ये आपसे ये उसका लिस्ट चाहिए था फर्स्ट ईयर वाला भी
So
Sir,
he's
Nehal, sir, you can continue. Nehal, sir, you can continue.
Yeah, he's MPharm PhD from Jadavpur University. He's having vast years of experience of 18 to 19 years. He's a recipient of Tamil Nadu Young Scientist Fellowship for the year 2015. He's a recipient of Young Investigator Award for the year 2015. And he's also recipient of Active Researcher Award. Sir has received many grants for conferences as well as travel grants. He has written book in his name. He is having more than 100 publications in his credit. He has presented 105 research papers in national conference, and in international conference also he has presented 30 research papers. He has guided postdoctorate students, two postdoctorate students, 3 doctoral research students, 44 MPharm students, and 54 BPharm students. Sir is also associated with APP branch, Tamil Nadu branch. Sir, it's my great pleasure to invite you on this platform to be an eminent speaker for today's 12th Indo-US conference, which has been organized by Umrak. So it's over to you, sir. Thank you. Thank you from my side, sir.
Good afternoon, everyone. Am I audible?
Yes, sir, you are audible.
Okay. So I thank you, Professor Nega, for your kind introduction, and thank you, the conference organizers, Professor Rajiv Dahiya, for giving me the opportunity. So let me share my screen and.
Sir, please share your entire screen.
Okay. Is it okay now?
Yes, sir. Yes, sir. It is visible.
Okay. So today, let me give you some updates about the biomedical applications of magnetic nanoparticles. So the theme of the conference is about nanotechnology, and you have got a very good introduction from the previous speakers and from other sources about nanotechnology. So our lab, we are contributing with the magnetic drug delivery. So let me give you a very brief introduction about nanotechnology, magnetic nanoparticles, and our own contributions on this particular area with respect to drug delivery. So this is an overview of my talk, and this you may be knowing that the definition, we need to handle around the particles with dimensions from 1 to 100 nanometers. So they are called as nanomaterials. It's very, very difficult, but people are working, and huge applications of nanomaterials and nanotechnology we could evidence nowadays from our day-to-day life. Most, most of the products are involving nanomaterials, and these nanomaterials may be naturally occurring or incidentally originated due to activities of humans, or otherwise specifically engineered by humans for specific applications. So huge number of nanomaterials are there, and these nanoparticles can be inorganic, polymeric, solid lipid nanoparticles, liposomes, dendrimers, nanocrystals, nanotubes. All these things are having pharmaceutical applications. Based on the morphology, it can be again classified based on the shapes, based on composition, based on functionality, based on surface modifications, the assembly. We can also classify nanoparticles. Still more technically, nanoparticles or nanomaterials can be classified based on its dimensionality. So 0 dimension, 1 dimension particle, 2 dimension and 3 dimension particles. So if you come to the 3-dimensional particles, mostly electrodes, all these things will come under the 3-dimensional category. And in 2-dimensional, it is paper-like materials or sheet-like materials like nanobells or nanoplates, composites can be there. Coming to the 1-dimensional, it can be tubes or it can be electrodes, it can be wires or it can be arrays. Coming to the 0-dimensional, it is core shell nanoparticle, encapsulated hollow particles or composite nanoparticles. So all these nanomaterials, they are having, I told you, dimensionality, the freedom to occupy a space. So the length, breadth and height, all these things can be occupied by the 3-dimensional particles, but for the 0 dimension, it is almost restricted. It cannot move any place. The dimensionality is totally restricted. So now this is the bulk and this is the quantum dot. 0-dimensional particle is called as quantum dot. Now this is the density of states, particular density of states for 3-dimensional, 2-dimensional and 1-dimensional, 0 dimension. And the relative magnetism can be visualized by these diagrams. So for quantum dots, the magnetism is on a very specific line, but here we can have a field. It is called as magnetic moment. So these magnetic materials, they have very unique characteristics. Very first one, that is the surface to volume ratio. So the bulky material is having a surface to volume ratio of 2, but the same nanomaterial will have a huge surface to volume ratio, that is 6. This is a gold. We know this is gold, very popular. The color of gold is also very known for us. But based on the shape and based on the size, the same gold will have different colors. So this is a spectrum of color, but it is all gold nanoparticles. So it is prism, red in color, 100 nanometers, spheres, 100 nanometers with more or less orange tinge, and spheres around 50 nanometer green color, spheres around 120 nanometer cream color, and so on, like this. These are the images. So peculiar optical properties for nanomaterials. Similarly, the bulk is having a very huge melting point, gold, but gold nanoparticles will have a very low melting point. So the thermal properties are also vary with respect to the nano scale. And this is a carbon, what we are using as a pencil, very soft material. This is called as graphite. The same carbon, when it is assembled in the form of diamond, it is much more harder. So the mechanically, it has huge changes with respect to normal bulky material and the nanomaterials. It has wonderful chemical properties when compared with the bulk and the nano dimensions. Sorry. Let me introduce magnetism. Actually, there was a huge rock, lodestone, which is nothing but magnetite, had a magnetic property which can attract nails. The reason is due to lightning. This rock was magnetized due to the lightning. Lightning carries a huge amount of electricity. Based on the electrical impact, this rock was magnetized, and it was a very basic observation by a shepherd. And the magnetism developed in the 2nd century BC by the Chinese emperors, and they have invented the compass, and it is highly useful for navigation. And the scientist William Gilbert, during 1544 to 1603, he made several experiments with the help of magnetism, and there was observation that even animals have magnetic property for their flying and returning back to their home. So we have permanent magnet, we have electromagnet, and as a whole, the Earth is also considered as a magnet because of its revolution or rotation around this axis. So magnet is described, the energy of magnet was described as a magnetic moment, and it is known as magnetization. Magnetization means how far a magnet can have a magnetic property, or a material can have a magnetic property. It is known as magnetization, which can be mathematically computed and described by two terms. One is permeability, the other one is susceptibility. So the magnetism can be classified based on the permeability and susceptibility. When the X value or the susceptibility value is very small and negative, then the materials were classified as diamagnetic. And the susceptibility is very small and positive, then it is paramagnetic. And when the susceptibility is large and positive, it is termed as ferromagnetic material. The examples were given here: diamagnetic gold, copper, silver, and so on. Similarly, aluminum, platinum, manganese are paramagnetic. Iron is known as ferromagnetic. So cobalt and gadolinium. So the behavior of magnetic materials with respect to magnetic field or field dependence. So the diamagnetic materials will have a straight line. It is curvilinear and it is a loop-like structure. So this loop-like structure is very important for our concept, which is known as hysteresis, and the property is known as ferromagnetism. And these magnetic properties were dependent on temperature, and the paramagnetic materials will have a parabolic nature of curve, and it is opposite for ferromagnetic materials. So pictorially, the paramagnetic material will have a zero magnetic domain because most of the magnetic moments will be there in different directions, and the net magnetic moment is more or less zero. But whereas in case of ferromagnetic, the magnetic moment is greater than zero, and antiferromagnetic, since it is cancelling each other, the magnetic moment is zero, and in case of ferrimagnetic, magnetic moment is greater than zero. So now these materials will have various critical temperature, the susceptibility value, the magnitude is various, and the temperature variation. So this is a comparison chart. And we are not interested in any of these materials, but we are very much interested in the super paramagnetic materials, and they are nothing but nanoparticles or magnetic nanoparticles because they have a blocking temperature and they have a very strong susceptibility, and the temperature is above the critical temperature, and it has a permanent dipole moment below the blocking temperature. So these are the unique properties of magnetic nanomaterials or magnetic properties. So then they were classified as super paramagnetic materials. So then there is one more concept called hard and soft magnets. So the magnets, this is a hysteresis structure, and the place of intersection at the x-axis is known as coercivity, and at the y-axis is known as remnants, and this is the origin. So when a particle is getting magnetized, so it starts taking this place and then it will follow this axis. So this is known as saturation magnetization, this point, and this is a microstructure that controls the coercivity. And the blue line indicates soft magnetic material. So here, this is a difference. So the magnetic property is totally different. So therefore it is termed as soft magnetic material. And if it follows a hysteresis, then it is known as hard magnetic material. Magnetic nanoparticles can be designed for specific application when they have a very single core inside them. So this is called as single domain, and now this particle contains many small particles, individual entities. They were called as multi-domain particles. So as a whole, this is known as hydrodynamic, and this is particle diameter, this is core diameter. So inside a core, there are small domains. So this is a multi-domain particle. Traditionally, we use multi-domain particles, but recently, specifically, we are having very good applications with single domain particles also. So we are manipulating the magnetic materials at the nanoscale in order to develop a single domain magnetic nanoparticles. So the multi-domain magnetic nanoparticles are permanent magnets, and single domain, they are exclusively nanoparticles or clusters, and they have a very good magnetic property or desired magnetic property when compared with the permanent or the bulkier magnets. So magnets and nanomagnets. So this is the demarcation line. So when the diameter is very small, means nanoparticles, and they are called a super paramagnetic, and this is a single domain, this is a multi-domain, and this bulk thing, and we are interested in this particular region. So single domain super paramagnetic nanoparticles for our pharmaceutical and other applications. So this I told you just now, coercivity, remnants, and saturation magnetization, and these are the other properties. Ferromagnetic will follow the brown line, super paramagnetic will follow the green line, paramagnetic will follow the blue line, and diamagnetic will follow the black line. So these are the magnetic moment and its alignment. So unblocked particles that respond to a field are known as super paramagnetic. Actually, so this image you try to see in half, means right side and left side. So when the magnetic field is 0, the particles will have their own magnetic moment in different directions here. But when you apply an external magnetic field, almost all the particles will align to the particular direction of the external magnetic moment. So this is called a super paramagnetism. So which occurs in nanoparticles, which are single domain and composed of single magnetic domain, and the diameter is below 3 to 50 nanometers. So they're mostly nanoparticles. Magnetic nanoparticles are having this property, super paramagnetic property will come under this definition. So now, why only nanomaterials will have this property? So this is based on the size effect. So here the isothermal magnetic remnant magnetization causes isothermal in the sense at a very constant temperature. I told you it is temperature dependent. So at various temperatures, the magnetic properties will behave differently. So this graph just I explained the single domain and multi-domain. So you could able to see visually this red line is super paramagnetic. This green will behave like this, and the blue line will have a larger hysteresis loop. So these magnetic properties of nanostructured materials fall under this particular category, the very small particles. So it is a size effect. The same materials will behave differently when the size is high. So now this red line, super paramagnetism, it passes through the origin. So therefore there is no remnants. Remnants in the sense residual magnetism will be 0. So therefore we prefer super paramagnetic nanoparticles for biological applications. So the next one, single domain and multi-domain. So this I told you, this is our range of interest. So their property or the angle of magnetization will look like this with the energy. So now based on these basic concepts, we can design magnetic carriers for biological applications so that we can have electrostatic repulsions, entropic repulsions, or any other requirement. It can be turned on or it can be turned off, magnetic moments. So we can design our magnetic carriers for biological applications. So all these things are multi-domain nanoparticles. It is microparticle. So we could able to tailor, we could able to modify this behavior, and we can have specific applications. So one of the prominent applications of magnetic nanoparticles is hyperthermia. Hyper means high, thermia means temperature, very high temperature treatment. So this is applied for cancer. So we could able to inject magnetic nanoparticles inside a tumor, and we can able to heat them externally, or we can control them remotely. So with the help of the heat that has been generated in the magnetic nanoparticles, we could specifically kill the cancer cells, and this is one of the proof of concept, and it is under clinical trials, and sometimes it is under human treatment in specific countries. But it is a very promising method of treating cancer. So the mechanism of heat generation of nanoparticles is when we are applying the external magnetic field, these domains will relax through two mechanisms. One is Brownian motion and other one is Néel relaxation. And based on these two processes, they start collide each other, and there will be friction between these two magnetic domains or multiple magnetic domains as a whole. And this friction causes generation of heat. It can be around 43 degrees Celsius. And when the temperature of is more than 40 degree or 41 degrees Celsius, the cancer cells will die immediately. So this Brownian rotation and Néel relaxation helps in establishing a very high temperature that is essential for killing the cancer cells, is called as magnetic hyperthermia. So the hysteresis loss will be here, means the magnetic loss will be there. So this will be the magnetic moment, and this is the hysteresis. So based on Neal relaxation and Brownian relaxation, the particles cause friction amongst itself, and this is the susceptibility, and therefore the cancer cells can be treated. So this is a relaxation behavior, Brownian relaxation and Neal relaxation. So the particles were turning itself here and there, so this causes friction. So this is the equipment for magnetic hyperthermia. So there is a coil, where induction coil, where magnetism can be generated, and this can be a mouse, and when it is exposed in the field generator through a fiber optic probe, we can measure the temperature. So these were reported studies. So that amount of temperature from a solid lipid particle or magnetic particle based on size, based on size dependent hyperthermia. So with various sizes, we could able to generate huge amount of temperature that is suitable for treating cancer. So this was the image from the reported article. So we could able to have a thermogram. So we could able to see the temperatures. So this is a biodistribution of magnetic nanoparticles. So very well it is located at the cancerous site or the tumor site. So therefore we may be able to specifically damage the tumor cells in the body. So this is a magnetically triggered drug delivery. So this is a gel. When electromagnetic field is exposed, gel network will release the drug, and we can have a drug release based on off mechanism, just like a remote. When you have an electromagnetic field, the drug release can be made on. When you remove the magnetic field, it can be made off. So pulsatile drug delivery is possible with the help of electromagnetism through the nanogel network. Similarly, magnetic particle imaging is a very new technique, and we all know that ultrasound or CT scan, topography, radio imaging. So all these things are having a lot of radiation effects, but this magnetic particle imaging doesn't have any side effects compared to other radiation techniques, and we can take images of blood vessels, we can take images of lungs, and many organs can be done. The scanning is like this trajectory, and finally it will be projected as a screen. It is a similar setup of CT scan, but it is having a relatively very less side effects and highly sensitive, very high penetration, no radiation at all. We can have a very good positive contrast, as well as we can quantify the data. So numerous advantages are there for the magnetic particle imaging. So these are the reported literatures for magnetic particle imaging when compared with MRI, and magnetic particle imaging gives a very good contrast, and therefore it is very well appreciated based on for the condition of GA bleeding. So these kind of bleeding images and all cannot be done through the radiation methods. Vascular imaging is possible only by magnetic particle imaging, and it can be coupled with isotopes and tracer techniques, so that we can have weighted MRI, means more amount of, so we can include the MRI along with fluorescence. We can have a color magnetic particle image. Usually it is black and white, but here we can have a colored magnetic particle images are also possible. We could able to differentiate organs based on the color contrast, liver, lungs, etc. So all these things are one of the best applications of magnetic nanoparticles. One more fundamental physics about nanoparticles or magnetic nanoparticles is the magnetoresistance. We all know resistance is nothing but friction. When two things are traveling in the same direction, there is no resistance. But when they are traveling in the opposite direction, there will be a resistance. So this is called as magnetoresistance, and giant means bulk, And when we are able to produce this giant magnetoresistance, it can be sensed, it can be quantified. So this particular concept is used for biosensors. So here ferromagnetic thin films and non-ferromagnetic conductors were placed, and usually the moments will be in the same direction, in B or C. But in case of this one, it is in the opposite direction. So when it is exposed to the magnetic field or the giant magnetoresistance sensor, and it can be quantified, and this magnetization or the resistance quantification is used as a sensor, and therefore they are known as magnetic biosensors. So non-ferromagnetic and switchable ferromagnetic. So here it is in the same direction, here it is in the opposite direction. It is switched on, so therefore we could able to generate resistance, and the resistance could be quantified. So that is the concept of sensor. So for the biosensor for food allergen detection, people have recently reported, and this principle, the use of magnetic nanoparticles. So the food and allergens along with the magnetic particle when exposed to magnet. So if there is allergens, then it will produce a detection. Within 1 minute we could able to sense. So this is the process. It is linked with IoT. So very well, easily we can detect whether the food is in a very good quality or not with respect to allergens. So this is one more sensor with similar thing for the sepsis diagnosis. So we could able to generate the or quantify the magneto resistance. It can be linked with IoT tools, and we can detect how much amount of sepsis is there. Sepsis is a kind of infection, long-term infection. And for infection imaging or infection diagnosis, so this is one of the very recent tool, and we could be able to do within 1 hour all this analysis we could be able to quantify. So these are hybrid magnetic nanoparticles, nothing but magnetic nanoparticles tagged along with radio isotopes. So they were known as radio sensitizers. So they have multiple applications. They're also used as sensors. So these kind of substances very recently reported for targeting the cochlea that is present in the ear. It is inner ear. It is relatively unreachable because of its architecture. It is a spiral anatomy. It has a spiral anatomy. So it is very difficult for us to reach inside the inner domain of the ear. So usually we will use a stylet or wire which will go up to this level. But these nanoparticles, very recently they have reported with the help of alloys and other things, so they could be able to reach the inner ear under magnetization. So these are the images which was recently reported in the year 2017. So the inner ear targeting with the help of magnetic nanoparticles. Similarly, brain, it is having a BBB, blood-brain barrier. So it is very difficult for us to reach the brain, but magnets can help the drugs through the nanoparticles to target brain and specific areas of brain. So this is the concept of brain targeting. So it was confirmed. And this one is for eyes. The bioavailability of drugs in the eyes is very, very less when compared to any other organ. So, but here the ocular magnetic micropumps will deliver drugs with the help of influence of magnetic field, applied magnetic field. So very good bioavailability was achieved with the help of the thing. So this was the micropump which contains a reservoir of drug. There is a valve, top membrane, layer, very small size, only 5 millimeter. So it could able to release the drug on an on-off mechanism. So we could able to see the pictures. This is a drug release. So with respect to time, that is, and with respect to magnetic effect, the volume of drug released is also changes. With very small magnetic field, small amount of drug is released. When you increase the magnetic field, more amount of drug is being released. So this is one another study of ocular micropumps. So could able to see the drug release with respect to time. So here there is an animation. It's not working, I think. So this ocular micropump insertion. So the micropump is inserted inside the eye through a microsurgery in a rabbit, and with the help of the magnet, it can be controlled using a device, and the therapy micropump implant site could be monitored, and it can release the drug, and it is highly appreciable. This slide is not working, sorry. So this was reported in the journal in the year 2018, Journal of Controlled Release on Demand VEGFR targeted drug delivery using magnetic micropumps. This is one another thing, ocular implants, and this paper is about lung targeting. The drugs could be specifically targeted in the lungs, and this is magnetic scintigraphy, means use of isotopes along with nanoparticles for imaging, scintigraphy, and it is useful for tissue grafting, so artificial skin, transdermal drug delivery, implants, oral drug delivery, many things, and even useful for water purification. Magnets could be used to purify waters. Most of the unwanted material in the magnets could be absorbed by magnetic materials and very easily removed by using an external magnet. So coming to our own contributions from our lab, so we have developed a magnetic nanoparticle based formulation for arthritis joints targeting. So this is a negative control and this is positive control with arthritis. This is standard drug, and formulation 2 and formulation 1, and magnetic drug targeting was there. So these were the X-ray images for various groups and various time intervals. Standard group, this is formulation 1 and formulation 2. So we have got very good number of publications with respect to magnetic drug delivery, and I thank the collaborators from Germany under the Indo-German joint research collaboration projects, and there is a huge number of scope or promise around these kind of drug delivery systems. So I told only magnetism. There can be light, there can be temperature, there can be pH, there can be chemistry, means redox systems. So the crosstalk can help us to develop multi-responsive system, and based on which we could able to improve the synergistic effect in the drug delivery system, and synchronous therapy is possible, and based on this we could have a very good pharmacology, and based on the improved pharmacology, so we can have multifunctional systems like the previous speaker told, theranosis. This is diagnosis, therapy, and therapeutic monitoring. So I think I have given you an introduction about magnetic nanoparticles and its very recent applications. Thank you all my students, collaborators, and thank you organizers, and thank you APP for the opportunity provided. So I'm happy to answer any questions. Thank you.
Okay, so thank you very much, sir. Now the session is open for question answer. Nihal sir, any question? Ajay sir?
Yeah, I have a question for Dr. Atinder, sir.
Yeah.
Yeah. Sir, both of you have nicely covered the topic nanotechnology and its therapeutic application. I have one a normal query. When we are talking about nanotechnology, it is something related with the better therapeutic efficacy of the particular drug. And when we reduce the size of particular powder or the particular drug, automatically it will increase the bioavailability as well as the therapeutic efficacy. But on the another hand, uh, it will I think it will also increase the chances of the side effect of the drug. So how we can manage or compensate both these things together, because on the one hand the higher therapeutic effect will be there, and on the another hand the chances of side effect will also be there. So how we can manage or how we can compensate these two things together?
Yeah, I mean, that is, I think, the at the same time advantage and one disadvantage is also there. So to counter
Yeah.
that disadvantage, we have to devise a strategy, and that is why targeted drug delivery is one approach, where
Yeah.
we target the drug to the desired site, not to the, and that is why maybe combination of all the concepts, like say our targeted drug delivery, the dosage form design, as well as then the stimuli response also, that we target to certain site and then we provide response stimuli, then only it is getting released, not until that, or we can control that its release or kind of that. Those type of aspects or the concepts or are being practiced, like people are trying, not 100% achieved, we can say, but that is where research is moving. So your idea, your question is very good, that if efficacy is increased, at the same time side effect also may increase. So to counter that, we have to devise counter strategies also.
And one more question for Dr. Palani Sami sir, Dr. P. Salva sir. Uh,
Yes.
we are talking about nanotechnology. The topic is very advanced and very novel and innovative. But when we talk about any drug or when we see the market, most of the formulations which are available in the market, they are basically the typical type of formulation or the normal formulations like the tablets and capsules or the ointment or the syrup. So, on the one hand, a lot of research is going in this area, and on the another hand, when we see the market, the normal or the traditional formulations are existing. So, if the topic is that much important, or if it is having that much basically, you can say capacity, why the basically most of the formulations which are available in the market, why they are not always the nano formulations?
Yeah, basically, this particular area is under developing very recently. Means from 1960 onwards, this concept of magnetism and its biological applications is underway. And in Germany, in hospitals, people are using for humans. I'm telling, they are using magnetism for treatment nowadays. So the German government has made regulations and approved, and it is allowed. But whereas other governments, they are still in the process of approval. Probably once if they approve, it may become popular like conventional dose. And we need to look into the benefit ratio of conventional as well as the magnetic formulations. whenever the benefits are high, then we need to go for a better formulation. Whenever the benefits are very low or the risk is very less, then we may go for conventional formulations. As a pharmacist, I can say. So this is where we are under development, and this particular area is proven nowadays in all animal models, cell culture models, but human models bit expensive, bit a lot of infrastructure is required and expertise is required. So that therefore it is under the primitive stage. Maybe in future it will be promising since it has a lot of advantages.
Okay. Thank you so much, sir. Thank you so much, sir.
Okay,
Now,
thank you.
yeah.
Thanks a lot.
Over to you, ma'am.
Thank you so much, sir, uh, for answering the questions. Thank you. I'm thanking you, Dr. Atindradi Shukla, Dr. Palanisamy Sedvamani, Dr. Ajay Sharma, and Professor Nehal J. Shah for sparing your valuable time for us. Thank you very much on behalf of Sri Nanjibai Lalbhai Patel College of Pharmacy, and thank you, all of you. I like to announce once again that Sri Nanjibai Lalbhai Patel College of Pharmacy has organized a free webinar for students of third and final year, how to crack GPET on 4th February 2021. We have also organized a free mock test for GPET on 30th February 2021, and the first 5 students who will secure the highest mark will be awarded with cash prize. Okay. So at this point, we have completed today's lecture sessions, and now it is time for break. After the break, we will continue with oral presentation at 1:45 p.m. onwards. So all the participants are requested to join us before 1:40 p.m. Thank you, all of you. Sir, anything you want to say? Uh, Dr. P. Selvani, sir, you want to say anything?
No.
Okay, thank you. Thank you for joining us. Thank you very much.
Innovation is power. The power to walk down never before known paths. The power to drive the transforming world today. The power to break the shackles of conventional and conquering it. We enable you to break the old. We are the Gujarat Technological University, the International Innovative University.
A proud alum. Hello everybody, I am Dr. Shivani Desai, a proud alumnus of Vidyabharati Trust College of Pharmacy, Umrak. I completed my B Pharm from Vidyabharati College in the year 2008. Later, I completed my post-graduation in clinical pharmacy from NMRS Mumbai and then PhD in the field of pharmaceutical sciences. With this background, currently I'm working as an assistant professor at Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune. Here, along with teaching the UG and PG students, I'm also involved in the research related to type 1 diabetes mellitus with many publications in indexed journals. To get over here, the basic requirement is the basic teaching. And the basic teaching I got from the teachers of Vidyabharati Trust College of Pharmacy. There I have learned all the basics of pharmacy as well as the basics of the life. The teachers over there just not educated us with all the technical and professional knowledge, but also the knowledge about how to lead a life. I'm really very grateful and thankful to all the teachers of Vidyabharati College of Pharmacy to get me over here in this life. Thank you so much to all the teachers and to the institute. Thanks. Have a nice day.
Everyone, my name is Rajvi Desai. It gives me an immense pleasure to talk about the institute where I had finished both my graduation and post-graduation. I had completed my graduation in 2009 and post-graduation in 2011. I feel privileged to be a part of the institute where I not only excelled in academics but also with the help and motivation of this inspiring faculty. A lot of good ethics and life learning skills, which can help which can help me for my better professional career. And also, the faculty of this institute is very knowledgeable, always ready and eager to lend their support to the students whenever is needed. Initially, when I had to start in 2005, I was very nervous being in an unknown environment. But as soon as I entered at this institute and I started, the atmosphere was very warm and welcoming, and I felt soon I felt very safe in this discipline institute, and where the help was always there whenever needed. And also, this institute takes a pride in providing the necessary resources to the students to study, which mainly I would focus on the laboratory aspect of very well maintained scientific instruments, which is like a great core to learn the pharmaceutical curriculum. And I had completed my post graduation in pharmacognosy, which mainly deals with the plant-based studies of naturally occurring active molecules, which can be used to treat various disease conditions. With if you see that plant has been widely used traditionally to cure the various disease condition with very less side effects. And so, and even the world's most nations has turned to the pharmacognosy for discovering the active molecules from the plants, which could be which could be very helpful to treat the various feared diseases, for example, cancer. So I feel so grateful to finish my graduation and post graduation from the institute, where I had all the resources all the time, and also the support and motivation from the faculties all the time, and all the help whatever I needed to finish my journey. It would not have been possible to finish this journey this easy and smooth without all the resources that I had from the institute, and also the motivation, help, knowledge, and support from the faculty. So thank you so much, and it was a great journey, and I am so thankful to be a part of such a great discipline institute, and being around the knowledgeable faculty. Thank you so much.
Hi, my name is Binoy Patel. I have completed my Bachelor of Pharmacy from Sri Naranji Bai Lalbai College of Pharmacy, Umrak, in 2012. I'm currently working as a compliance validation specialist in Celgene Pharmaceutical at New Jersey location. What makes this College of Pharmacy special is sense of belonging. I can see where it's like a big family. It's a student focus and all professor, principal, lab supervisor, and each and every staff member was very helpful, supportive, and encouraging during my 4 years of college life. I think learning pharmacy has not to be content and classroom style, but it has to be real lab experience. That's what I have learned and gained during my 4 year of bachelor degree. The faculty and staff was great. They care for the students and they want them to be successful in their career. We had a great celebration of each and every festival, days, and each and every festival throughout the all 4 years. I had made so many great friends, and we still keep in contact with each other. Same as we have a great bond with the all professor also. I would like to thank you all for giving me the opportunities and brightening my career. Thank you very much.
Innovation is power. The power to walk down never before known paths. The power to drive the transforming world today. The power to break the shackles of conventional and conquering it. We enable you to break the old. We are the Gujarat Technological University, the International Innovative University.
Hello everybody, I am Dr. Shivani Desai, a proud alumnus of the Vidyabharati Trust College of Pharmacy, Umrak. I completed my B Pharm from Vidyabharati College in the year 2008. Later, I completed my post-graduation in clinical pharmacy from NMRMS Mumbai and then PhD in the field of pharmaceutical sciences. With this background, currently I'm working as an assistant professor at Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune. Here, along with teaching the UG and PG students, I'm also involved in the research related to type 1 diabetes mellitus with many publications in indexed journals. To get over here, the basic requirement is the basic teaching. And the basic teaching I got from the teachers of Vidyabharati Trust College of Pharmacy. There I have learned all the basics of pharmacy as well as the basics of the life. The teachers over there just not educated us with all the technical and professional knowledge, but also the knowledge about how to lead a life. I am really very grateful and thankful to all the teachers of Vidyabharati College of Pharmacy to get me over here in this life. Thank you so much to all the teachers and to the institute. Thanks. Have a nice day.
Hi everyone, my name is Rajvi Desai. It gives me an immense pleasure to talk about the institute where I had finished both my graduation and post graduation. I had completed my graduation in 2009 and post graduation in 2011. I feel privileged to be a part of the institute where I not only excelled in academics but also with the help and motivation of this inspiring faculty. I learned a lot of good ethics and life learning skills, which can help which can help me for my better professional career, and also. The faculty of this institute is very knowledgeable, always ready and eager to lend their support to the students wherever is needed. Initially, when I had to start in 2005, I was very nervous being in an unknown environment. But as soon as I entered at this institute and I started, the atmosphere was very warm and welcoming, and I felt very safe in this discipline institute, and where the help was always there whenever needed. And also, this institute takes a pride in providing the necessary resources to the students to study, which mainly I would focus on the laboratory aspect of very well maintained scientific instruments, which is like a great core to learn the pharmaceutical curriculum. And I had completed my post graduation in pharmacognosy, which mainly deals with the plant-based studies of naturally occurring active molecules, which can be used to treat various disease conditions. With if you see that plant has been widely used traditionally to cure the various disease condition with very less side effects, and so, and even the world's most nations has turned to the pharmacognosy for discovering the active molecules from the plants, which could be which could be very helpful to treat the various feared diseases, for example, cancer. So I feel so grateful to finish my graduation and post graduation from the institute where I had all the resources all the time, and also the support and motivation from the faculties all the time, and all the help whatever I needed to finish my journey. It would not have been possible to finish this journey this easy and smooth without all the resources that I had from the institute, and also the motivation, help, knowledge, and support from the faculty. So thank you so much, and it was a great journey, and I am so thankful to be a part of such a great discipline institute, and being around the knowledgeable faculty. Thank you so much.
Hi, My name is Binoy Patel. I have completed my Bachelor of Pharmacy from Sri Naranji Bhai Lalbhai College of Pharmacy, Umrah in 2012. I'm currently working as a compliance validation specialist in Celgene Pharmaceutical at New Jersey location. What makes this College of Pharmacy special is sense of belonging. I can see where it's like a big family. It's student focused and all professor, principal, lab supervisor, and each and every staff member was very helpful, supportive, and encouraging during my 4 years of college life. I think learning pharmacy has not to be content and classroom style, but it has to be real lab experience. That's what I have learned and gained during my 4 year of bachelor degree. The faculty and staff was great. They care for the students and they want them to be successful in their career. We had a great celebration of each and every festival, days, and each and every festival throughout the all 4 years. I had made so many great friends, and we still keep in contact with each other. Same as we have a great bond with the all professor also. I would like to thank you, all of you, for giving me the opportunities and brightening my career. Thank you very much.
His voice really impaired.
Innovation is power. The power to walk down never before known paths. The power to drive the transforming world today. The power to break the shackles of conventional and conquering it. We enable you to break the old. We are the Gujarat Technological University, the International Innovative University. Innovation is power. The power to walk down never before known paths. The power to drive the transforming world today. The power to break the shackles of conventional and conquering it. We enable you to break the old. We are the Gujarat Technological University, the International Innovative University.
I'm working as an assistant professor at Dr. D.Y. Patil Institute of Pharmaceutical Sciences and Research, Pimpri, Pune. Here, along with teaching the UG
A very good afternoon, ladies and gentlemen. I am Mrs. Pooja Chaudhary, Assistant Professor at Sri Naranjibai Lalbhai Patel College of Pharmacy, Vidyabhati Trust, Umrak, wholeheartedly welcoming you all again in 12th Indo-US International Conference. I hope you all are safe in this pandemic. Before starting the session, there is an announcement. Shri Naranjibai Lalbhai Patel College of Pharmacy is going to organize a webinar on 4th February 2021 on how to crack GPAT and on 13th February mock test for GPAT 2021. A complete test for all the subjects will be organized. I am providing registration link in the chat box. Registration is free of cost and cash award will be given to top 5 students. For more details, visit our college website. Now, without further ado, I will turn the time over to oral presenters. Our first oral presenter is Dr. Madhu Gupta, and the session is being chaired by Professor Y. Shravan Kumar. At this moment, I take an opportunity to introduce Professor Y. Shravan Kumar. He presently serves as professor at Vagadevi College of Pharmacy. He awarded PhD degree from Kakatiya University in 2011. He completed M.Pharm in the year 2005 from Bharati Vidyapith Deemed University and B.Pharm in 2002 from Vagadevi College of Pharmacy, Kakatiya University. Professor Y. Shravan Kumar has 16 years of experience in teaching and research. He guided 60 M.Pharm and presently guiding 6 PhD students. Professor Y. Shravan Kumar has published more than 60 articles in peer-reviewed national and international journals and authored different book chapters. He is acting as reviewer for international scientific publications as a current drug delivery, drug metabolism, drug interaction, etc. His research interest includes novel drug delivery system, drug-drug and drug excipient interactions, and BABE studies. Professor Y. Shravan Kumar attended and presented posters in several international and national conferences. He evaluated the poster session in the 15th Indo-African Conference. He also awarded for Best Poster at Indian Pharmaceutical Congress. He is a life member of Association of Pharmaceutical Teachers of India. He received DST and CSIR travel grant for attending AAPC conference in New Orleans 2010. He awarded with Best Teacher Award presented by Association of Pharmacy Profession APP in Indo-African Conference 2019. He attended many QIP programs as resource person. He also attended 10th and 11th Indo-US International Conferences and 66th IPC as evaluator. I feel honored to invite our esteemed chairperson, Professor Y. Shravan Kumar. Over to you, sir.
Organizers for giving me this opportunity. Is it audible? Yeah. I thank organizers for giving this opportunity as a chairperson in Indo-US 12th Indo-US conference. The title is going to be Nanotechnology in the Pharmaceutical Sphere, Contemporary Approaches and Therapeutic Applications.
Hello, good afternoon to everyone. Hello, so sir, shall I start?
Yeah, start.
Okay, so first I share my presentation. So sir, is it visible?
Yeah.
Okay. So a very good afternoon to everyone. Myself Dr. Madhu Gupta and I am working in Delhi Pharmaceutical Sciences and Research University as an associate professor. Today I am going to present the topic that is entitled as Polymer Lipid Hybrid Nano Vehicles that is designed for synergistic drug delivery for solid tumor management. So first of all, my major research area that is basically focused on the pharmaceutical nanotechnology and as well as the targeted drug delivery. I have interested and published several formulation based publications that is used for the bioligands for targeting of bioactives, drug moiety, biopolymers, cancer nanomedicines, as well as the topical fungal infections, psoriasis, wound healing management, and several other topical delivery related formulations. So first of all, today I have presented on the topic is that is solid tumor management. So here my presentation is basically focused on the first we have discussed on the recent updates on the solid tumor therapy and their management. And the next, how we can overcome the challenges that is associated with the pathophysiology of solid tumor and how we can exploit these challenges to the opportunity and how we can prepare a drug delivery system that is most suitable for the management of tumor therapy. And third part is focused on the to discuss the research work that is done on the topic hybrid nano engineered system that is designed for the synergistic drug delivery for solid tumor management. Now here I just focus on the top 10 cancer related research news. Fat clogged immune cells that can ultimately fail to fight the tumors. And recently FDA approved a drug that is target the key genetic driver of cancer. Third important news is the inhibiting protein that could enhance the immunotherapy of effectiveness. Fourth important point is the obesity that can set to overtake smoking as biggest preventable cause of cancer. And recently, all of we know कि Allison and Honjo got awarded a Nobel Prize for the field of medicines. And the next important point in related to cancer, that is the survival rates, that is double for patient with melanoma brain metastasis that receiving the immunotherapy. And that is one of the most important thing that is I explored in the internet, that cancer cells always greedy for the glucose. And recently there is a delivery system that is based on the advanced type of delivery system, nano robots, that can seek and destroy the tumor. So ultimately these are the 10 important news related to cancer research. Now I would focus on my presentation. So first of all, what is the basis of research proposal that I have selected? As all we know, in tumor cells as well as the normal cells, both have the different types of pathophysiology. So the abnormal microenvironment of solid tumor, such as tumor vasculature, hypoxia, and pH. All these play a key role in terms of solid tumor physiology, and ultimately that is have a great impact for designing of the novel drug delivery system that can ultimately use for the targeting of the particular site. So these are the two pictures that can be show, an A point and B pictures. A picture showing the normal tissue and the B picture showing the tissue that is having the cancer or tumor. So the next slide is the most important thing is the receptors. The receptor that is overexpressed on the tumor cells as well as the tumor vasculature. As all we know, in the tumors there are several types of in cancer disease there are several types of receptor that is overexpressed on the tumor cells. But here I have selected the CD13 receptor, or we can say APN receptor. Why we have selected? Because such type of receptor, it is overexpressed on the tumor cells as well as the tumor vasculature. So the main purpose of this research is to the synergistic drug delivery, because when we design a such type of nanoparticles that is having the ligand that is uh, having a ligand that is can easily interact with the CD13 receptors, and it can easily kills the or target the tumor cells as well as tumor vasculature indirectly. We all of we know that one tumor vessels have nourished the thousands of tumor cells. So if we directly kill that one tumor vasculature cell, that means indirectly we can kill the thousands of tumor cells. So ultimately it provides such type of delivery system will provide the synergistic drug effect. So now this is a time to know the basic information about the APN or CD13 receptors. So aminopeptidase N, or we can say APN, or the CD13, that is known as the cluster differentiation protein. It is a cell surface based receptor, and it is highly overexpressed at the endothelial cell surface, in the neoangiogenic blood vessels and the tumor cells, and it can contribute in binding as well as involved in the angiogenesis of tumors. This receptor is a membrane bound zinc dependent metalloproteinase and the most important ligand that can easily target the APN receptors, that is the NGR peptide sequence, and the sequence is the asparagine, glycine, arginine. It is a most popular ligand that can be used for the APN receptors. So now what is the basis of drug delivery or the carrier that we have selected? So we have selected here is the polymer lipid hybrid nanoparticles because it provides the benefits of both lipidic nano carriers as well as the polymeric based nano carriers. So we have selected this polymer lipid hybrid nanoparticles. It can provide the higher drug load. It has the capacity to the longer systemic circulation. It provides the controlled release profile, and it is able to incorporate both type of drugs, hydrophilic as well as the hydrophobic molecules. And if we go to this molecular structure of this particular PLN, or we can say polymer lipid hybrid nanoparticles, so it is having the PLGA core around with this lipid monolayer shell of lecithin and a PEG linked lipid that is used for the binding or you can say conjugating the ligand. So what is the basis of selection of targeting approach? So we have selected a nanocarriers that is having the PEG surface. So the PEG is a hydrophilic type of property, and it can provide the escape from RES uptake, and it provides a longer residence time in blood circulation. So we have selected such type of nanoparticles, and it can provide the controlled release of drug, and the drug is released in controlled manner, so the chances of dose dumping is reduced. So what is the role of NGR ligand on the APN CD13 receptor to recognize them? So this is a nano carriers that is having the NGR ligand, and whenever the ligand binding nanoparticles goes into the systemic circulation where they reach into the target site, and they can easily recognize the CD13 receptors and bind them. But in case of normal cells or healthy cells, this type of nanosystem are not able to recognize the normal cells, because normal cells don't have a CD13 receptor. So such type of delivery system is not killing cytotoxic effect for the healthy cells. It ultimately used for killing the tumor cells. So this can be used. So what is the basis of dual targeting delivery of solid tumors as well as tumor endothelial cells? So this is a graphical representation. In this graphical representation, we can see if a person or if a patient that is having a solid tumor, if it is directly administered or IV administered nanocarriers, designed nanocarriers, and it ultimately goes into the systemic circulation where due to the EPR effect, they are reached into their target site. And in the target site, this picture clearly show their green color showing the tumor cells and this red color showing the tumor endothelial cells, and both type of cells having the CD13 receptor. So the design system can easily target both sides, tumor cells as well as tumor endothelial cells. And in our ex vivo study, we have taken a model for tumor cells. We have taken the HD1080 cells, and for tumor endothelial cells, we have taken the model cell Hubeck cells. So the ex vivo study is done on both type of cells. So this is the uptake and processing of nanoparticles inside the cells. If we consider the case of normal cells, when these ligand anchored nanoparticles reach into the normal surface of the normal cells, where the normal cell does not represent the cell receptor, so they are not recognize the receptor, and ligand receptor interaction, it does not take place, so no cytotoxicity will take place. But in case of tumor cells, when such type of targeting system reached into the surface of tumor cells where the CD30 receptors is overexpressed, and where ligand and receptor interaction takes place, and ultimately such system goes into inside the cells where they fuse with the lysosome and form the endolysosome, where the hydrolytic enzymes takes place and the washing is done, and drug is released into the cytoplasm, and ultimately it targets the microtubule assembly. So here in this case, I have taken the paclitaxel as a drug. So now this is the hypothetical approach or our planning. Now, what is the aim of this study? We have prepared the synthesis and characterize the first kg NGR modified PLG lecithin nanoparticle, PLG lecithin PEG based copolymer. Then we prepared such a pre-designed copolymer into nanoparticles. Then we have performed the ex vivo study on the both tumor cells lines, tumor cells as well as tumor endothelial. Now the main objective of this study is to achieve the proof of principle for the hypothesis that the designed nanoparticles can increase the anti-tumor effect by improving the penetration of drug in solid tumor cells as well as increase the therapeutic efficacy. So again, we have prepared the nanoparticle on the basis of emulsification solvent evaporation method. So this is the schematic representation. Now we have gone to the characterization part. So the nanoparticles showing the approximately 170 nanometer particle size.
Sorry to interrupt you, ma'am. You have 5 minutes to complete. You have
Okay,
five.
okay, okay.
Thank you, ma'am.
Okay, so the targeted nanoparticles showing the around 82% efficiency and the conjugation surface density is 198. So this picture is showing the apoptosis study, and this study is showing ki how the drug-induced nanoparticles can easily kills the tumor cells or not. So this A part is on the HT1080 cell line and B part showing the UVEX cells. And we can find ki targeted nanoparticles is easily segmented the nuclear part and form the apoptosis body because their chromatin part is easily segmented. So this type of nano system can easily able to kills the tumor cells. This is the their percentage apoptosis cells. This is the cell line is in vitro cytotoxicity study that is done on the 24, 48, 72 hours and shown that targeted nanoparticles having the least or very lesser require the very lesser dose for killing the tumor cells. This study is showing the cell cycle analysis, and here we have taken the this study was performed on the tumor cells, and here we can say the four partitions are present: P1, P2, P3, and P4, and these P2, P3, P4, and P5 that is represented the various phases of cell cycle, and we can found in our experiment ki that such type of nano system is target the G2M phase and it can arrest the G2M phase and it can having the higher density because cell cycle arrest in this phase, particular phase. This type of cellular association of various types of nanoparticle formulation that can be presented on the tumor cells, and here ABCD four types of pictures is showing, and specifically if we focus on the C and D, C showing the targeted nanoparticles and D showing the targeted nanoparticles, but such type of cells is pre-incubated with the free ligand. So we can find in the picture C, maximum nanoparticle can easily enter into the nucleus or can easily enter the inside of the cells. So the fluorescent intensity is brighter. But in case of D, if the cells is first pre-incubated with the target ligand, then ligand is reached inside the cells, then there is very less amount of receptors is available for interaction of the nanoparticles that is conjugated with the NGR ligands. That's why here intensity is very lesser. In vivo study is performed on the Wolf C mice and we have taken 4 groups. So this study is showing the in vivo anti-tumor efficacy study, and we found that ligand conjugated nanoparticle that is KNGR based, it is having the property to tumor volume that is very less as compared to other formulations. So it can we can say the tumor volume density it can kills the most of the tumor cells, or it is able to manage it can easily manage the tumors. It can easily manage for it can be easily used for the solid tumor therapy management. And if we consider the toxicity of this nanoparticles, this study can be considered by the percentage body weight change. So if the animals that is treated with the targeted nanoparticles, they doesn't or very negligible body weight change, so we can say that this formulation is considerably less toxic as compared to the other groups. If we see the drug, different types of drug loaded formulation on the tumor. Yes, sir.
We are
I
unable to
think.
see the other slides.
Yes, sir.
The only first slide we are able to see from that time.
Oh, sir, you are seeing only first slide?
Yeah. You are not changing the slide.
Sir, but I have seen the my slide number 25 in this. I don't know ki how it is happened. Can I again share?
At least from now, what you are seeing, please try to share.
Ma'am, please select entire screen.
Yes, ma'am. I have used entire screen. Yes. Now it is able to see.
Yes, it is visible.
Yeah.
Is it visible?
Yes, ma'am.
Yes.
So shall I continue, sir?
Yeah.
So I am sorry for this because I don't aware ki you are not able to see the my screen. So this study, I'm continuing my part. So in this study, that is showing the percentage tumor volume growth percentage inhibition and percentage tumor weight growth percentage inhibition, that can represent by the percent VI and percentage WI. In both cases, we can see this column is the highest because the targeted based nanoparticles is able to higher the percentage growth, tumor volume growth inhibition and percentage tumor growth inhibition. So we can see such type of delivery system can be a remarkable approach for the solid tumor management. This graph is the percent survival of the different group formulation treated groups, and we can find in our study outcomes that PLN KNGR, or we can say targeted nanoparticles treated group, having the longest survival span period, that is around to 55 days, but in other formulation it is less. So we can say ki yes, such type formulation treated group having the longest survival span period. So now the summary and conclusion part, we can see ki, the such type of dual targeted nanoparticles in which we have only conjugated one type of ligand, but it is able to target both sides, tumor cells as well as tumor endothelial cells. So we have by using this approach, we can mm, uh, we can do the or we can say ki, the complexity of the carriers we can reduce, and by simplifying the nano carriers we can easily target both sides, tumor cells as well as tumor endothelial cells, because there are so many publications they are targeting only the tumor cells. But if we are able to target the tumor endothelial cells as well as other than tumor cells, so one tumor endothelial cells is targeting, if it is kills, then it kills indirectly thousands of tumor cells. So basically this is our concept of the whole this research project. So the some finally it can significantly, the nanoparticle that is designed, they can significantly enhance their specific uptake by both cells, tumor cells as well as tumors endothelial cell, by following the CD30 receptor mediated endocytosis. The enhanced intracellular uptake that is presented by various studies. So the results of overall studies supported that such type of nanoparticles easily enhance the intracellular uptake of targeted nanoparticles that can directly result in stronger induction of apoptosis, and they can produce the greater cell cycle arrest that is done on the G2M phage level. This is the cell cycle stage, and the in vivo results were also found to be concurrent with the ex vivo results, and the anti-tumor efficacy studies as well as their survival.
Sorry to interrupt you, ma'am. Ma'am, you are running out of time. We have to conclude it. Thank you.
Okay, okay. So it's now from my side, it is completed. Okay. Yes, ma'am.
Professor Y. Shravan Kumar, sir, you have to tell something.
See, I have one question that you have performed these experiments in mice.
Yes, sir.
Okay, then might be the expression of your what we call proteins or no enzymes. They may not be similar to that of mammalian species.
Human. You can say ki
Yeah,
human. Sir,
yeah.
yeah, it may be because I'm not the expertise of that biochemical part, but it may be possible कि the internal body system of the mice and the humans is not exactly similar, but this is just a model, so it gives some outlines of this proposed system. Yes, it is can be used, or it is provide the some therapeutic efficacy in, or if it provides, to in which extent it can provide. So it just out giving some outlines before proceeding with this, or we can say if we want to commercialize this product, then of course we have to go for clinical trials and other steps, because it is a lab based study. So if we go to the next level, then surely we follow the guidelines of CDSCO.
I mean to say that there should be something like allometric scaling where you project from rodents to the mammalian species. Okay.
Yes, sir.
So another question what I have was you are saying lipid nanoparticles. So did you address the problem of coexistence of colloidal particles?
Sir, actually, we have prepared the polymer lipid hybrid nanoparticles, not lipid nanoparticles exactly. So it has the properties of both polymer lipidic polymer nanoparticles as well as the lipidic nanoparticles. So it may be chances, because it is a nanoparticles or it is a nano system. So we have performed also the SAM and TAM study, but I have not included in this part. So in my salmon study, I am not finding. We have just found around 170 to 200 nanometers range of particles. It may be the vesicular structure, because it is a chances, and we have not go for pilot level, so maybe chances. But overall, this design system provides a good therapeutic approach and good results. But overall, actually my voice is echo. Yes, sir. Uh, Hello, sir. You are not audible. Uh, Sir, you
I
are
think
not audible.
no.
Yes.
I think it's audible now.
Yes, sir.
Yes,
Yes.
sir.
Okay. So I had only two questions that because when we are preparing a lipid nanoparticles, we may get another carrier structures also, like even
Yes,
message
sir.
also.
It may be chances, but in my SAM or TAM study, we don't find such type of other structures, because we are also pop up the SAM and TAM. But in case of whole lipidic dispersion, it may be chances, but we don't having any such type of study that can differentiate between the both nanostructures.
And the only point of concern is about the liquid purity. Mainly when you try to procure, might be you have procured from the lipoid company. About the
Lipid
phosphoric result.
purity.
Yeah.
Yes, sir. Sir, we
Yeah.
have egg PC used, and that is procured from the Sigma Aldrich. That is having around 99% purity. So
Means the only company used commercially is going to be the lipoid
no,
company,
no,
which
no,
is
no,
which is
sir. There are so
generally
many
in the
company.
company. No,
Lipoid
the company's
also.
what? Yeah. So
Yes, sir.
which shows the minimum toxicity.
Yes, sir.
When we go for commercial scale, we can go for using of that.
Yes, sir. Sure, sir.
Okay, all the best.
Thank you, sir. Thank you. It's a nice suggestion, sir. It's a nice suggestion. In future, actually, it's my PhD work. If we having go to for next level, then surely I will consider your advice. Thank you, sir.
Okay.
Thank you very much, sir. Thank you very much, ma'am.
Thank you, ma'am. Thank you.
So the next candidate is there.
Oral presenters
Any
are
question?
requested to conclude their session within 15 minutes. Thank you. Dr. Jagruti Desai, currently working as Assistant Professor, Ramanbhai Patel College of Pharmacy, Charusat University, Changa, is second presenter, and the session is being chaired by Professor G. Rajitha. It's my honor to introduce Professor Rajitha. She serves as Professor and Chairperson of Board of Studies, Institute of Pharmaceutical Technology, Sri Padmavati Mahila Vishwavidyalaya, Tirupati. She was awarded the PhD from SPMVV, Tirupati in the year 2008. She completed her B.Pharm and M.Pharm from Kakatiya University in the year 1992 and 1994 respectively. Professor G. Rachita has 23 years of experience in teaching and research. She has published more than 45 articles in peer-reviewed journals and authored different book chapters. She is a reviewer in many reputed international journals. Under her guidance, more than 40 M.Pharm and 5 PhD students were awarded. Professor Rajitha is a member of various national professional bodies including APP, IPA, IPTI, ISTA, etc. and fellow member of Indian Association of Pharmaceutical Scientists and Technologists, Calcutta. She also a editorial board member in International Journal of Pharmaceutical Science and Nanotechnology, International Journal of Pharmacy and Biological Sciences, and Journal of Human Pharmaceutical Sciences. Professor G. Rajitha's research area includes synthesis of heterocyclic compounds, hydrazones, acyl hydrazones, and cinnamides. He is a recipient of 60 lakhs rupees grant from DST and research project. He also completed 3 major projects under UGC with 14 lakhs rupees grant. Professor Rajitha received Best Poster Award in 66 IPC. She also received award in 4th International Women Award by Venus International Foundation as a distinguished woman in health and medical science held at Chennai. It is an honor to have you here, ma'am. I find this a matter of great privilege to invite Professor G. Rajitha. Over to you, ma'am.
Yes, thank you. Am I audible?
Yes, ma'am, you are audible.
Am I audible? Yeah.
Yes, ma'am, you are audible.
Thank you. At the outset, I would like to at the outset, I would like to thank the organizing members for giving me opportunity to act as a chairperson in this nanotechnology 12th Indo-US conference on nanotechnology in the pharmaceutical sphere. Thank you. Thank you for all the organizing members, especially APP President Rajiv Dahiya sir. Thank you very much. You can proceed, Jagruti Desai. You can continue
Yes,
your talk.
Yes, ma'am. Good afternoon.
You can share the slides.
Yes, ma'am. I have shared the slide. Is it visible, ma'am?
Yes, ma'am. Yes, ma'am. It is visible.
Yes, now it is visible. Now it is visible. Presentation mode. You just keep presentation mode, no?
Yes, I have done the presentation
Full
mode.
slide,
I
full
think,
screen.
yes, I have done the full screen now. I hope it is full screen in others windows also.
For me, it is not there.
Is it
Yes, it is visible.
okay, but not the full screen. Just a minute, let me do it again. Now,
Yes. Yes.
it is full screen.
Before starting, I want to introduce Jagrati Desai. She is
yes, ma'am.
Presently working in Ramanbhai Patel College of Pharmacy. And earlier she worked in Sardar Patel College of Pharmacy and Pioneer Pharmacy Degree College. And she was selected as a She is the co-author of the paper awarded Professor M.L. Corona Memorial Prize Best Paper for the year 2013 in the field of pharmaceutics and biopharmaceutics. She guided five young project students. She received Charusat Research Grant on development of oral lipid-based formulation of anti-neoplastic agents for enhanced lymphatic uptake, and she received this grant in the year February 2019. With this brief introduction, you can start. You can start the presentation mode.
Yes, ma'am. Thank you for the introduction. And
I welcome Jagrati to start.
Yes, ma'am. Am I audible? Am I
Yes,
audible?
yes, ma'am, you are audible.
Yes, I have done the presentation
Yes, you
mode.
are
I
audible.
think. Okay, ma'am. Very good afternoon. Myself, Dr. Jagruti Desai, currently working as assistant professor at Ramanbhai Patel College of Pharmacy. So today I'm going to present on enhanced oral bioavailability and lymphatic targeting of darunavir using nanocarriers. Briefly, what is HIV? It is human immunodeficiency virus. It is a lentivirus that causes AIDS where the immune system begins to fail. So in 2013, around 35 million people lived in the HIV AIDS, and until 2013, there were around 39 million deaths. So then HIV heart therapy was introduced in humans. This HIV virus, it affects the T cells. Now these T cells have CD4 receptors on their surface. So this human retroviruses, it is divided into two categories. One is human T cell leukemia virus, that is HTLV1 and 2. The other one is human immunodeficiency virus, HIV1 and HIV2. So this is HIV1, which is basically. Yes, ma'am.
Jagrati, sorry to interrupt you.
Yes, ma'am.
Your slides are not moving.
Oh. Just a minute, ma'am. Let me try it again. So, is it now introduction slide? Is it now introduction slide?
Yes, ma'am.
Yes.
Okay. Limitation of the heart therapy, slide number 3. I hope limitation of the heart therapy, slide number 3 is there.
Only first slide I am able to see. I don't know
Okay.
about others.
Yeah, same.
Now, is it the third slide?
Title slide I am able to see.
Oh.
Not slide number 3. Yeah, now the second slide, introduction.
Actually, I have already removed it.
Yes,
Maybe.
yes. You can
Now, is it third slide?
Yeah. Now it's the third.
Yes,
Yes.
miss.
Okay, I'm not doing the full screen because there's some technical problem in the full screen where I am able to change the slide, but it is not getting changed. Okay, so limitation of the heart therapy. This heart therapy, it requires lifelong treatment, and around 95% of the adherence to this treatment is required. Patient incompliance is observed because of the high doses of the drugs required. Then poor drug targeting to the reservoir sites. These reservoir sites are the organs in the human body where this HIV cell resides and replicates. So the conventional therapy is not able to reach these reservoir sites. Then the long-term treatment is there. It is lifelong treatment, so it is obviously related to the adverse effects, the drug-drug interaction, then cost effects also. Also, many of the drugs like proton pump inhibitors, they are the substrate for the efflux transport, and because of which their bioavailability decreases because they get effluxed in the GI tract. So I have selected the drug darunavir, which is a protease inhibitor. Its molecular weight is 547, and it acts as a HIV aspartyl protease, which the virus needs to cleave the HIV polyprotein into its functional fragments. Log P is 2.82 and PK is 2.39. The oral bioavailability is 37%, and in markets, the oral tablets are available 75, 150, 300, 400, and 600 mg tablets are available. This tablet, that is 600 mg of darunavir, is generally administered with 100 mg of ritonavir because ritonavir is administered in order to prevent the metabolism of the darunavir. Then water solubility, very high solubility in water as ethanol at form at 20 degrees Celsius, and it is a substrate of P glycoprotein which is found in the cells. Then I have selected the lipid-based drug delivery system because the unique properties of lipids like the physicochemical diversity, the biocompatibility, and its proven ability to enhance the oral bioavailability of many drugs. Because of this, then many water-insoluble drugs and lipophilic drugs are being administered by this lipid-based drug delivery systems, and they are also administered through the selective lymphatic uptake. Lymphatic uptake by reducing the efflux transport activity at the intestinal cell wall. The lipid-based formulations can increase the solubility and the dissolution of lipophilic drugs and facilitate the formation of the solubilized species from which the absorption occurs and thus can reduce the food-dependent bioavailability. I have selected the solid lipid nanocarriers as a lipid-based drug delivery system for darunavir because it gets additional advantage that it is proven to be taken up by the M cells, which are present in the gastrointestinal epithelium cells, and it will enhance the lymphatic uptake. So also the peptide, which is been developed by the GenScript Picatsoe, that peptide was grafted on the surface of the solid lipid nanoparticles. This particular peptide has the affinity to bind to the CD4 receptors of the T cells. That is, it has affinity for CD4 receptors, and CD4 receptor bearing T cells are the target cells for the HIV. So ultimately, I have selected that peptide, and after preparation of solid lipid nanoparticles, that peptide was grafted on the surface of nanoparticles. So before that, the drug loaded, that is darunavir loaded solid lipid nanoparticles were prepared by emulsification solvent evaporation technique. The drug and the solid lipid, it was dissolved in dichloromethane. Earlier, these 6 to 7 solid lipids were screened: combritol, then placirol, then HCO, that is hydrogenated castor oil, dinosine 112, 114, and 116 were taken. Then, based on the entrapment efficiency and the particle size, the hydrogenated castor oil was selected as the final solid lipid. So the drug and the solid lipid was dissolved in dichloromethane. Then this organic phase, it was homogenized into the aqueous phase, that is sodium oleate solution in water. Then the emulsion, the so formed, was then sonicated using the probe sonicator, then followed by evaporation of the organic solvent at the room temperature for around 3 to 4 hours. Then the nanoparticle dispersion which was formed, it was centrifuged at 10,000 rpm, 10 minutes at 4 degrees Celsius using the sigma centrifuge. So upon this formulation and optimization, the peptide was grafted on the surface of solid lipid nanoparticles. The peptide sequence mentioned in the slide was grafted on the surface. Then for this grafting, 100 microgram of the peptide and SLN equivalent to 1 micromolar of the lipid was taken, and it was combined in 100 microliter of the buffer, that is 0.9% NaCl and 10 millimolar NaCO3, and it was done at pH 6.5 at room temperature. Then agitation for around 8 hours in an incubator. Then to remove the unbound peptide, nanoparticles were dialysed using 1 liter of the buffer, and then the nanoparticles were lyophilized using trehalose as a cryoprotectant. So the equation given here is a chemical reaction where the peptide, that is HS bond, because of this sulfide bond, it binds to the surface of the nanoparticles, and then this kind of linkage is formed. So characterization, the particle size was found to be 194 nanometer, done in zeta size and zeta potential of minus 35.45 millivolt. Entrapment efficiency was found to be 90.10. For entrapment efficiency, the nanoparticles were centrifuged at 1000 rpm for 10 minutes, and then the supernatant was collected. The settled portion consists of the unbound drug. So supernatant was collected, and then the drug estimation was done, and it was found to be 90%, while the drug loading was estimated to be 13%. Transmission electron microscopic image, this confirmed the shape as well as the size, which was in accordance with the particle size analysis that was done. Then the in vitro drug release study. In vitro drug release study was done for 2 hours in simulated gastric fluid, and then rest of the 10 hours in simulated intestinal fluid. So this is the, just a minute. Then the qualitative determination of surface grafted peptide, that is, was performed. For this, the composition of resolving gel and the stacking gel is given here. This method was taken, which was available in as SDS page study. The method was not modified as such. The whole method was adopted. This is the qualitative determination, whereby we can see that in the figure there is a non-reducing page and a reducing page. In a non-reducing page means DTT was not added. DTT is ditriethylal. That means it has the capacity to break the sulfide bond. So once the sulfide bond is break, the peptide can give the band. So 1, 2, and 3 is the lane for this peptide solution, not nanoparticle solution. So it was giving the band, and in non-reducing page there was no band found at 4, 5, 6, means DTT that is in peptide formulation. DTT was not there, so it did not cleave the sulfide bond, and the band was not obtained. In reducing page where DTT was added, 1, 2, 3 is the standard peptide solution, and 4, 5, 6 is the formulation which that is in the reduced condition. Because of breakage of the sulfide bond, the peptide was free and it was able to give the band. So this is qualitative determination. Quantitative determination was also done by Laurie-Bronsted assay method, where 0.5 ml of the sample was taken in Eppendorf and the solution, that is the formulation, was added upon dilution with a buffer. Then 0.7 ml of the Laurie's solution was added and it was vortex and incubated in dark at room temperature for 15 minutes. Then upon incubation, again the Foley reagent was added and a vortex, and again it was kept in dark for 30 minutes. Then after 30 minutes, the samples were analyzed colorimetrically at 660 nanometer wavelength using UV visible spectrophotometer, and the percentage conjugation was calculated. Standard was adopted as BSA standard calibration curve was taken. So percentage conjugation was calculated. This is the percentage conjugation assay BSA standard calibration curve that was obtained, and the percentage conjugation was found to be 73.12%. This is the graph for the in vitro drug release study, where the peptide loaded peptide grafted solid lipid nanoparticles of darunavir and simple darunavir loaded solid lipid nanoparticles. There was insignificant difference between these two graphs, and which confirmed that the loading grafting of peptide on the surface of an SLNs, it had no effect on the drug release from the SLNs. Around 20% release was found in around 12 hours from both the nanoparticles. Then confocal microscopic study was also done. 6-coumarin was used as a fluorescent agent, and it was loaded in the SLNs, that is peptide grafted and non-peptide grafted.
You, ma'am. Sorry to interrupt you, ma'am. You have few minutes to conclude this. Thank you.
Okay, ma'am. Okay, so MALT4 cell line, which expresses CD4 receptors, and simple CACO2 cell line, which doesn't express the CD4 receptors, were used in this confocal microscopy. The procedure for confocal microscopy was as available in the papers. So this is the image for the MALT4 cell line and the CACO2 cell line. In MALT4 cell line, A image, that is DAPI staining, which stains the nuclei. Then second one is B image, that is it stains the cytoplasm, where the formulation can be seen. And C image, it is the merge of A and B image. Then in CACO2 cell line, similar images were obtained. Now comparing both these cell line images, we can see that in MALT4 cell line, the green fluorescence is high, which says that the more amount of formulation was present. So this is the confocal study. Then in vivo pharmacokinetic studies were done in male vista rats, where after kinetic study, the samples were analyzed using the LCMS technique, which was developed, and then using the kinetic software, non-compartmental model was used. This is the graph that is obtained, Cmax of the formulation and peptide loaded formulation 452.31 nanogram per ml was obtained. Then Tmax of around 1 hour was obtained. With this, the bioavailability in comparison to the drug, in comparison to the marketed formulation, it was found to be around 150%, that is 1.5 fold higher as compared to the marketed formulation. Yes, 117% relative bioavailability was increased in comparison to darunavir suspension, and for darunavir loaded nanoparticles, the relative bioavailability increased by 481.35%. So in SLN, it was observed that Tmax was quite increased by 1 hour in comparison to marketed tablet, where it was found to be 0.5 hours. Then lymphatic transport study was also done, where cycloheximide was used, which has the capacity of blocking the lymphatic system, lymphatic circulation. So in that, when it was administered intraperitoneally, this was the effect that was observed, that in presence of cycloheximide, the amount of darunavir SLNs was found to be low. That means that the darunavir loaded SLNs were taken up by the lymphatic system. So in conclusion, darunavir loaded solid lipid nanoparticles were successfully prepared, and it was loaded, and the peptide grafted was successfully done on the surface of nanoparticles, and it was qualitatively and quantitatively determined. 73% conjugation was also observed. Then in pharmacokinetic study, the bioavailability increased, and in lymphatic transport study, we were able to find that the darunavir loaded nanoparticles were successfully getting taken up by the lymphatic system. So these are the references. Thank you.
Yes, Jagruti, am I audible?
Yes, ma'am.
Yes, you have done very good work. I congratulate you. Actually, what you have done, darunavir, is actually water soluble compound, no? It is a hydrophilic
No,
in
ma'am.
nature.
No, ma'am. It is lipophilic in nature.
Lipophilic in nature, but in the physicochemical properties, you said it is hydrophilic.
No, ma'am. It is lipophilic in nature.
Achha, okay.
It is poorly water soluble, not totally insoluble. It is poorly water soluble drug.
Water soluble. So among all the, marketed formulation tablets is there, suspension is there, but yours is nanoparticles, protein loaded.
Yes,
Which one is better?
In context of?
In all the contexts, kinetics path and uptake.
I have I have seen that the SLNs which I had prepared, I think without even peptide grafting, it was able to increase the bioavailability. So maybe peptide grafted nanoparticles were, of course, they were able to reach and they were able to bind more to the lymphatic system. So when we want that the drug to reach to the reservoir site, we can go for peptide grafting. While for everyday therapy, this kind of SLN, lyophilized non-peptide grafted, can be converted into lyophilized powder and it can be filled up into a capsule. So that can be also a better option.
Oh, very good. Yes, actually, your work is very good. Have you communicated this?
Yes, ma'am.
No, you have accepted, paper is accepted?
No, ma'am.
You have just communicated?
Yes, ma'am.
Okay. To which journal?
A European Journal of Pharmaceutics and Biopharmaceutics.
Okay. Very nice work. Actually, I'm a little bit layman in this field. My part is medicinal chemistry, but still I could follow you. You have very well presented.
Yes, ma'am.
Thank you.
Thank you, ma'am. Thank you, ma'am.
All the best.
Thank you, ma'am.
Dr. Jagrati?
Yes, ma'am.
Thank you very much for your nice presentation and sharing your research with us. I have a question about one thing, that is, this is to address the poor bioavailability of the anti-AIDS drugs, right? So you have shown the KCO2 cell lines. So do you think the mechanism of the enhanced bioavailability, is it only the nano size, or do you also claim that nanoparticles could have changed the permeability, or it is only due to the cellular uptake of the nanoparticles?
Yes. The work which I have present is a part of a bigger work where I had prepared 3 nanoparticles of different sizes. One was less than 100 nanometer, the other is less than 200 nanometer, and the third one is around 500 nanometer. And then the pharmacokinetic study up to in vivo study, I had compared all the 3 nanoparticles, where I was able to see that the results of 100 nanometer particles and 200 nanometer particles were non-significant. but it was having greater bioavailability in comparison to 500 nanometer particles.
Yes.
So this was the observation that I had obtained, and to conclude from that, I had observed that 200 nanometer is sufficient. We need not to decrease it to less than 200 nanometer.
Okay, because for the KCO2 cell line, the purpose was to check the cellular uptake, not the permeability, right?
Yes.
Okay. And did the KCO2 cell line studies give you any idea about the bioadhesion of the nanoparticles? Because when we administer the nanoparticle by oral route, then we suppose that because of that nano size, they have a better adhesion to the mucosal surface of the GIT. So, can you have any clue from your KCO2 cell line studies? Or
I had also done the intestinal permeability study using this CACO2 cell line, but that study is not mentioned in this presentation because of time limit. So in that, I had observed that the nanoparticles were able to more retain on the CACO2 cell line, but it is quite primitive, and I am not well versed with the cell line studies, so it is difficult to answer.
Yes, because in the literature we usually do not find information about the permeability change. We often found about the what you say is the cellular uptake because of their smaller size. Okay, and you already mentioned that if you want to make a final dosage form, the fabrication of the nanoparticle could be in form of capsules rather
Capsule,
than tablets.
yes.
Okay,
Yes.
thank you.
Yes, ma'am. Thank you.
Thank you very much, ma'am. Our next presenter is Ms. Renu Saharan. Ma'am, please try to complete before 3. Ms.
Okay,
Renu
sir.
Saharan presently serves as Assistant Professor in Department of Pharmaceutics, College of Pharmacy, Maharishi Markandeshwar Hind University, Ambala, Haryana. The session is being chaired by Dr. Sunita Dahiya. I would like to introduce Dr. Sunita Dahiya. Dr. Sunita Dahiya presently serves as Assistant Professor of Industrial Pharmacy at School of Pharmacy, University of Puerto Rico, USA. She completed her PhD from Gautam Budh Technical University, Uttar Pradesh, M.Pharm in Pharmaceutics from Rajiv Gandhi University of Health Sciences, Karnataka, and B.Pharm from AR College of Pharmacy, Gujarat. Dr. Dahiya has more than 70 years of experience in teaching and research. She has published several articles in peer-reviewed journals and authored different book chapters. She is a reviewer in many reputed international journals and editor-in-chief of Scientific News magazine, published by Association of Pharmacy Professionals. Dr. Dahiya's research are include Dr. Dahiya's research area includes dissolution enhancement of poorly water-soluble drugs using various approaches, as well as development of controlled release formulations by polymeric dispersion techniques. Dr. Dahiya is a recipient of seed money grant from National Institute of Minority Health and Health Disparities from the National Institute of Health. Dr. Dahiya's academic interests include active learning methodologies such as problem-based learning, classroom seminar, journal club, small group workshop, students e-portfolio, and rubrics creation of the assessment. Dr. Dahiya serves as General Secretary of Association of Pharmacy Professionals since 2011 and is fellow member of various national professional bodies including APP, IPA, APTI, IPGA, etc. and international bodies such as American Association of Pharmaceutical Scientists and American Association of Colleges of Pharmacy. We rarely get this kind of opportunity to invite a torchbearer person, Dr. Sunita Dahiya. I respectfully invite Dr. Sunita Dahiya to chair the session. Ma'am, please.
Thank you for the nice introduction. And our next presenter for the oration is Ms. Renu Saharan. you have already introduced, but let me tell you that Ms. Renu Saharan is presently pursuing her research PhD degree from the Banasthali University, Rajasthan, and she is presently working as assistant professor in the Department of Pharmaceutics at College of Pharmacy, University, Mulana, Ambala, Haryana. I came to know that Ms. Renu Saharan is going to deliver her talk on delivery of drugs and biomolecules using graphene sheets. So I think in today's all the sessions, this is the first talk about the inorganic nanoparticles, and we hope that everybody will learn and everybody have something to take home about the, because our conference is about the nanotechnology, and this is the only session which addresses the use of the inorganic material, that is the carbon, in the drug delivery area. So I invite Ms. Renu to deliver her talk. Ms. Renu, over to you.
A very good afternoon to all. Am I audible?
Yes, you are.
Okay. First of all, I would like to thank the organizing committee to give me such an opportunity. So today I am going to deliver a presentation, and my topic of presentation is delivery of drug and biomolecules using graphene sheet. So these are the content of my presentations. A brief introduction about nanotechnology, then pharmaceutical nanosystems, the type of carbon materials, graphene and its property, functionalization of graphene and its application in medical field. So as we all know that nanotechnology is basically deal with the small structures having size 0.1 to 100
Ms.
nanometers.
Renu, we are not
Yes,
able to see your slides.
just a minute.
Ma'am, please select entire screen.
Is it visible, ma'am, now?
No. It seems that it is in your email. I think I can see the first slide, a small picture of your slide on your email.
Yes, ma'am, but I have opened it.
I think you just close down all other windows and just open the presentation and then share the screen so that you will get the permission to share your slides.
Ma'am, I have opened it in my window, but it is not visible to you. I don't know why.
I don't know, any other person can see, because I can't see anything.
No, ma'am, it is not visible.
Okay.
Just a minute, ma'am. I'm again sharing.
Ma'am, first open your PPT. After that, you will find share screen option on your screen. And then select entire screen.
Is it visible, ma'am?
No. Yes, yes, now it is visible.
Okay. So this is about nanotechnology that in this we study the creatures or structures which having size zero point 0.1 to 100 nanometers. And the term was coined by Norio Tanguchi, and Dr. Richard P. Feynman was the father of nanotechnology. He is known as the father of nanotechnology. These are the various fields in which we have applications of nanotechnology. These are the energy sectors, medicines and drugs, nanotechnology, nano devices, optical engineering, defense security, bioengineering, cosmetics, and fabrics. As we know that the field of nanoscience has grown much over the last 2 decades, and the importance of nanotechnology has increased in all these areas. So in medical fields, many pharmaceutical nanosystem have been already developed. Like here we can see that these are the examples of various pharmaceutical nanosystems. First is the drug conjugate. Nanosystem basically the combination of two or more components for any therapy or cancer treatment, etc. So here are the example of these pharmaceuticals nanosystem. Here first is drug conjugate, then missiles, dendrites, quantum dots, carbon nanotubes, metallic nanoparticles, etc. So, and these are the various pharmaceutical nanosystems. So, the nanocarriers in drug delivery. Nanocarriers actually belong to the nanoscales and used to deliver the drug. They are basically designed to control the drug release, enhance permeability, biological membrane, change the drug distribution in vivo, and improve its efficiency. So these are the basics, some nanocarriers that are nanoparticle, vesicles, nanosheets, and nanocapsules. But today I am dealing with this carbon nanomaterial. This is one of the most interesting element, as the majority of part of living as well as non-living world is made up of carbon. So these crystalline carbon exist in these following states that are diamond, graphite, fullerenes, carbon nanotube, and graphenes. So there are different type of dimension-based structures that here we can see. This is zero dimensions that are the clusters, fullerenes, and diamonds. One dimension that is carbon nanotube, and two-dimensional structures is of graphene sheet. So nowadays graphene, this is considered as a wonder material of 21st century due to its brilliant physico-chemical properties, which we are going to discuss further. So first we should know what is graphene. Graphene actually is a single layer of graphite. This is a pure crystalline carbon which was isolated by the professor Andrew Game and with his former students Konstantin Siloy in 2004, and they also got the Nobel Prize for their discovery. So now, what are the properties which bring graphene in this limelight? These are the that it is quite thinner materials. Here we can see that its thickness is around 0.345 nanometers. This is the strongest material ever measured. It has a good electrically conductance. It is very light in weight. It conducts heat even better than diamonds, and it has tunable electronic properties. It is very flexible and stretchable, transparent too. So, in field of biomedical, uh, our concern properties are like it has a very large surface area, have good mechanical strength, and high surface ratio. So these are the properties which make it a very useful materials in the field of biomedicines. But the thing is that we can't use graphene as such because it has very limitations. Here we can see that graphene itself is a hydrophobic in nature, and it also consists of so many impurities. For this, we have to functionalize the graphene first, and we can functionalize it as a in the form of graphene oxide and reduce graphene oxides. So after converting this graphene into graphene oxides, we can create the oxygen group on it, or epoxide groups and carboxyl or hydroxyl groups on it, due to which it became hydrophilic. That can be used to enhance the solubility of many hydrophobic drugs. Then this reduced graphene oxides, it is actually an intermediate between the ideal graphene sheet and the oxide graphene oxide. So as I have told that the graphene is a hydrophobic material and it requires modification of the functional group to make it a biomedical material. This modification we can achieve by the covalent or non-covalent functionalization of the graphene sheet. So by functionalizing it, it will get converted into graphene oxides, which is hydrophilic in nature, and we further can modify the surface of this graphene oxide sheet, which is now hydrophilic in nature. Sorry, by using some biocompatible polymers like chitosans, polyethylene glycols, etc. So by using such polymers, we can also make it a biocompatible. This functionalization also improve the dispersibility, biocompatibility, binding.
Ranu ma'am,
Yes,
Ranu
ma'am.
ma'am,
Yes, ma'am.
you have to complete within 5 minutes as guests are
Okay,
gathered
okay,
for
just
the question. Thank you.
okay, ma'am. So we can achieve this functionalization by in two ways, that is covalent and non-covalent. So by functionalizing here, we can see that first we created the groups on graphene sheet and it will convert into graphene oxides. But after that, we can functionalize it with various agents here like imaging agents, drug, and molecular targeting. So by molecular using targeting moiety, we can achieve the targeting drug delivery. Imaging agents, they are used to create bioimaging. So this whole thing will create a nano systems for the better drug delivery of any therapeutic agents. So here we can see that how this functionalization of nanographene oxides is achieved. Then various applications of graphene nanomaterials in medical field. Here we can see that graphene and graphene oxides have its applications in various fields like tissue engineering, mechanical strength, mechanical conductivity, optical transmission, biological properties, and nanobio electronic device. But um, our concern area is medical fields, where we can improve the therapeutic index, viability, or solubility of a drug by using these graphene oxide sheets, by using them as a carrier or a conjugate with these drugs. Here, the source of these graphene oxide-based drug delivery vehicles is depend on the three factors. That first factor is constructing a carrier with an optimum loading capacity. Second, to confirm the degree of toxicity and biocompatibility. And third is to design a system that able to release a drug in a controlled manner. So by functionalizing these graphene oxide sheets, um, these we can develop graphene based vehicles which can release the drug in acidic pH. So this is by using the pH sensitive polymers. There are many other chemical strategies also by which we can create the controlled release of drug by using these sheets. Here, this is the anti-cancer function of the graphene-based nanomaterials. Here, this is a anti-cancer drugs. By using these graphene sheets, we deliver these drugs to the target area. So in two manners here we can see that how graphene sheet is using by loading anti-cancer drug on it, or by using infrared light by which it get photo activation there, and by using heat they can act on the cancerous cells. So it has both properties, can act either by loading drug on it or by using infrared light. So it is an example. Here we can see that this is graphene sheet which have converted into oxide form, and by using the polymer, here is a PEG. Then we use to load drug on it, and by adding a target moiety on it, it release the drug at the target site. So this is the approach which we can follow to design a drug delivery system. Here are the examples in which these graphene nanosheets act as a composites to deliver the drugs, and here the highlights of the studies, that how they can enhance the or improve the delivery of these drugs and their actions. So I'm not going to read each this. So here are some genes also and biomolecules which we can deliver using these graphene oxide sheets. As we have seen that the functionalization of these nanographene oxides by different methods like covalent functionalization and non-covalent functionalization, or decoration of these sheets with the polymers. They show excellent features in medical field. The polymer functionalization graphene can offer stronger interaction actually, which can enhance the load transform from matrix to graphene. But one thing is here is that to understanding of the behavior of graphene and its derivative in biological system, which is not only important to investigate their toxicology but also to create or to improve their performance in various biomedical therapies. So here we can conclude that the by using functionalized graphene is in triggered release with external stimuli such as pH, magnetic fields, or an near infrared radiation. It is fast emerging as a novel gene delivery platform. But the in vivo fate of these graphene carriers after local or systemic administration did not receive enough attention till now. Actually, few studies have also done in some past years, but still there is a lot of things pending which we have to include, like their toxicity, their bioavailability. Some literature shows that they are biocompatible up to some extent, means their toxicity depend upon the dosage, their dosage. So the future emphasis on tissue distributions, mechanism of clearance and toxicity is required to understand their true potential. So these property may have a significant effect on the safety, toxicity, and clearance of these materials upon administration. Knowledge of in vivo behavior of different graphene family materials will pave the way of expanding their application in drug and gene delivery. So it is expected that the synergetic effect from scientists in different areas include like material science, biology, or clinical medicines may promote further exploration of graphene-based material in bioscience. So we can say that this is the um, that by using these graphene oxides as a nano carrier, we can improve the biocompatibility, biodistribution, and solubility and tumor targeting efficiency of many chemotherapeutic agents, as it has capability that it has a high loading capacity due to enormous large area, ease of functionalization by interaction, or remarkable physico-chemical properties also. So in this review, I just included the approaches by which we can use these graphene oxides as a nano carriers for many therapeutic or chemotherapeutic agents, and included many examples which researchers have already done with these graphene oxides. So thank you. It's all.
Thank you, Ms. Renu, for your presentation.
Thank you, ma'am. Thank you for this
It gave a lot of
opportunity.
information about the graphene sheets as a nanocarrier, especially the carbon-based nanocarrier. I have a question that this is carbon-based nanocarrier, right?
Yes,
Uh,
ma'am.
Before graphene, we have heard about the carbon nanotube. So
Yes,
what
ma'am.
exactly make graphene sheets different from the carbon nanotube?
Ma'am, first is that it is two-dimensional structure. So first is that it has a high loading capacity, and second, its biocompatibility or its colloidal property is greater than carbon nanotube. At that we can generate maximum carboxylic or oxygenated group on it. So it has more stability than carbon nanotubes, which I have came to know from literature survey.
Okay. Okay, and I think in case of graphene, or whether it is graphene or whether it is carbon nanotube,
Yes, ma'am.
the effect of the shape as well as the aspect ratio is very important on the
Ye-
toxicity, because in last you mentioned about its in vivo behavior.
Yes,
Before,
ma'am.
yes,
Actually, in vivo behavior is still a question mark to the scientist. Actually, it's a toxicity. Some are saying it's dose dependent, uh, and it's has a high loading capacity as compared to carbon nanotube.
yes. Is there any specific method for loading the cargo in the graphene sheet, especially the graphene oxide?
Ma'am, there are two ways that we can simply loading the drug by non-covalent bonding, pi stacking, or we can use some linker like some biocompatible polymers or synthetic polymers between these graphene sheets and the drug moiety.
Okay, okay. Thank you very much.
Very much well. Thank you very much. All the best to is joining 12th INDO-US International Conference. Now, for validatory functions, the thing which is known as work polymer. To validate all asking results, now you are requested to join Google Meet for validatory function, which is given to your WhatsApp group. Thank you very much, all of you. You may leave.